Norway spruce, the first two talks. And the last talks will be looking at different parasites affecting pines, so spruce and pine for this morning. The first is Blessing Rudola from the University of Göttingen.
Good morning, everyone. Because of the day, I hope to start the day on a good note. And there's some people already listening to this talk in this point, so just take it as another opportunity to listen to Blessing again. I'll be talking on heterobacterium and the impact of genotypes on the response of schools to this pathogen. Okay, so I give a brief introduction about Heterobacterium. It's a species complex that has several species within it, and they are commonly found in Eurasia and also in North America. So, this pathogen has its own distinct hosts. They prefer certain hosts, but they also are able to attack other hosts, and the distribution follows. The distribution of the pathogen, is that me or?
No, I'm sorry.
Okay, so the distribution follows the pathogen distribution follows the distribution of the host, as you can see. So in Europe and Asia, we have Heterobacterium sensu, we have Heterobacterium, we can do next.
You can maybe try have a look yourself.
Okay. It was off.
Okay.
We have Heterobacterium calvicorum that protects food. We have Heterobacterium allotum sensu, and that protects P. and Heterobacterium aviatum, which protects Avis aurora. While in North America, we have Irregularia and Occidentalia, and they protect Avis, Unipax, and value, we think it's going to be billions already. So briefly about how it spread, Heterobacterium produces both sexual and asexual spores, but from literature, the primary mode of infection in nature is through the basidiospores. And the spores are produced on the fruiting body, also called the basidiocarp, and then they are carried by wind. They land on freshly exposed surfaces. It can also be through wounds on the stems or on the roots, but the primary mode of infection in the nature is through stumps. So these freshly corn stumps, the Heterobacterium infects this, establishes itself, and then begin to spread to other living plants around it. So we can mostly detect the decay in the stem and also in the roots. So several symptoms are visible, two of which I'll mention is the pathogen species involved and the tree species infected. Like I mentioned earlier, these pathogens have their favorite host, but they also attack other hosts. In spruce, for instance, at least in Europe, the fungal colonization happens in the heartwood. And when this happens, there are no visible symptoms on the outside. And when the tree is weakened by the pathogen, when there is heavy wind throw or felling, then the extent of the damage is visible. The most effective control method for this is removing the stumps and all the roots, but of course that is cost-intensive and also laborious. So there are chemical agents used, and also this controlled fungus, Phleboopsis gigantea. But as we know, both chemical and biocontrol agents are not even acceptable practices in a lot of countries. So planting trees that are less susceptible to this pathogen would be more effective. And that brings me to the aim of our study, which is to determine what factors are responsible for the resistance and susceptibility of Norway spruce, to 2 different strains of Heterobasidium parviporum. Inoculate borer to bore through the stem to reach the surface of the sapwood, and then we place equal-sized plugs of either strain of Heterobasidium or agar, which was our control, on the exposed surface, and then we sealed with paraffin. Then we left it for certain number of weeks. So in total, we had 222 plants, each for both strains, also control, and then we left 134 plants totally untreated. So these plants were distributed into 2 watering groups. So we had some that received optimum watering, while the others received half of the watering received by the 100% group. Then we measured the diameter, the height, and the growth of the plants. So in total, we had 800 plants, and they belonged into 8 families and 82 clones. So the combination of this was what we used as the genotypes. So how we measured the necrosis, we gently scraped the bark, to reveal the lesion in the phloem, and then we further scraped to see the extent of the damage in the sapwood. So we measured the length and the width of the lesion for both Heterobasidium treated and the control plants. And obviously, the non-treated plants showed no visible sign of lesion, so nothing was measured there. So briefly to the results, from the 5th week of inoculation, we observed significant differences between the 2 treatments. But these differences did not affect the height growth of the plants. So the plants grew pretty much the same. The only difference we observed was in the diametric growth. So the plants from the normally watered treatment grew better in diameter than the low water treated plants. And also we examined based on different families and different genotypes. So we have, and these 2 figures are for the height growth. So we observed that family 48, for instance, grew better than all the other families, but it was significantly different compared to 42, 47, and family 50. And the genotypes also, there were lots of combinations, and there were significant differences amongst some of them. So for the necrosis, we measured necrosis in the phloem and in the sapwood, and we measured that both for the horizontal and vertical directions. And there were no significant differences across all, except, and in the sapwood, oh God. Okay. So much for the first talk of the day. Okay, so only the lesion width and the phloem had some significant differences. So the pathogen benefited from the drought and then caused greater necrosis compared to the normally watered plants. And then we tried to see the effect of the inoculation on the lesion sizes. And for all the treatments, there were significant differences. But for the phloem and the sapwood, there were differences amongst all the treatments in the lesion length. So control was significantly different from both strains of Heterobasidium. But for the lesion width, for both phloem and sapwood, there were only differences between the control-treated plants and the 2 strains, but there were no differences between among the strains. So we concluded that HPA2, which is the second strain, caused greater necrosis compared to the first strain, although in general, there were significant HPA2 grows faster than HPA1. Then we tried to do a correlation between the variables. So for figures A and B, we correlated between the lesion length and the lesion width in both phloem and in the sapwood. The higher the lesion in the phloem, the higher it is in the sapwood. Then we tried to correlate the starting height with the lesion width, and there were weak to moderate correlations between them. Also, we correlated the diameter with the lesion width, and there were moderate correlations between them. So we say that the greater the diameter, or the larger the diameter, the larger the necrosis, but on a moderate scale. So to summarize this talk, again, we try to see what factors are responsible for the growth and resistance of different families and basidion, different watering availabilities. So the first result, we observed that the fungal strains define their virulence among the spruce genotypes, although the height growth was not affected by the inoculation methods. And also, the growth of the plant varies between the genotypes and the families. Again, the height growth was not disturbed by abiotic stress, and we think that is due to the low drought stress that the plants were exposed to, and also the season in which we did the experiment. So it spanned through the winter, and of course the plants don't need so much water at that point. So we think it's important to consider how different plants react to different pathogenic strains, because this is important when developing biocontrols or selecting resistant genotypes for breeding purposes. So to the projects that we are currently doing, together with Anna, we are trying to see what fungal communities are in some of these plants. And the first method we are trying to use is the ITS2 sequencing. Then we collected some of this material 16 weeks after they were inoculated. So in total, we used 85 DNA samples. 67 were from the phloem and 18 from the root samples. Of course, we would have liked to, sequence all the 800 plants, but no money, right? So we extracted both the total RNA and the genomic DNA. We are going to use the RNA for differential gene expression later. And the second method, together with Tanya, we are trying to use the traditional-based cultural method. So the same 18 root samples that were used in the previous step, we sterilized and plated them, and we have, this amazing-looking fungi that we characterized morphologically, and then we came up with 40 groups. Then we tried to extract DNA and sequence from each group, and we are using different primers also to validate, to see if we will have the same results, from different primers. So for now, it's still ongoing anyway, but we had 82 OTUs, and we've been able to identify 61 of them with the ITS markers, and 58 were also confirmed by the LSU. And then we would compare the results from this traditional culture-based method with the metagenomics. And yes, that is it for my talk. I would like to thank my supervisors, Eva and Catherine. And also, it's a big project. A lot of people are involved. But I would like to thank the German Ministry for Education and Research for funding this project, and also my thesis committee members, Professor Geiling and Professor Asiebu. And like I said, in our group, we are not only, meat lovers. I said that in Lisbon. We sometimes change our lifestyle like pests also, and we are vegetarians, so don't mind the huge pile of meat. And with that, I would like to say thank you all for listening, and thank you.
Very, very well timed. Thank you, Blessing. That was actually 13 minutes, so you had time to spare. But thank you for that talk. The next speaker is Malin from Uppsala. Will also be talking to us about Norway spruce and Heterobasidium. Thank you.
So where is a good distance for the microphone now? Let's see if we can get this down to an art. Okay, so we will continue with the topic that Blessing started and talk about the interactions between conifers and different strains or genotypes or whatever of Heterobasidium. This is the work of, you really don't see the screen, let's try this instead. So this is the work by one of my former students, Edan Capador. And he likes to play on words, so hence the very catchy title, The Firm Tree Does Not Fear the Storm. I'm not really sure we could prove that. And now let's see if I can change. Yes. So as Blessing introduced, there are various species of Heterobasidion in Europe, and in southern Sweden we have 2 of them in symmetry, meaning we have both the broad host range Heterobasidium anosum in the strict sense, and the more narrow host Heterobasidium parviporum, but they both readily infect spruce. And that means that the forestry and the breeders have to take these 2 in to make big of a difference. But as Blessing discussed, this is not the case. So for Hernan's thesis, trying to get this down, our sort of overarching question was, will a tree that's resistant to one pathogen species also be resistant to the other pathogen species, or to one isolate to the other isolate? If you look first at the scatter graphs to the right, this was something that almost did us in when we did this graph. This is the resistance in a set of 269 families for both Heterobasidium parviporum and Heterobasidium anosum. And you can draw the line anywhere you like in this cloud. There is no correlation that we could prove. But what we could find was that the genetic component was rather strong in both experiments, and we could use the data and identify 19 loci which were pleiotrophic. It meant that they were controlling resistance traits for both species. Sometimes they were synergistic, so same allele would confer resistance to both. Sometimes they were antagonistic, but there is at least some genetic component going on here. That sparked our interest in what's going on really. What is the trait robust? Or are we looking at genotype by genotype interactions? So basically for his 4th chapter of the thesis, and this is still work in progress, Anand asked 2 questions. In a uniform environment, will a spruce genotype respond? And how much of that variation, if there is variation in response, will be controlled or reflected in the gene expression of the partners? So we really went back to basics. We have one population in Sweden which accidentally was tested for Heterobasidium resistance in the field. So this is an old experiment where one of the repetitions in the clone trials were planted on a very misfortunate site where about 50% of the trees were rotted really early, even in the rotation time of 25 years. But as you see, there is very So we wanted to use that material for artificial inoculations, and Blessing really nicely explained the inoculation system, so I'm not going to spend very much time on this. We do slightly different, we have small differences. We actually measure an additional trait to the trait that Blessing measures, and that is the spread of the pathogen in the sapwood. But this talk is actually not on that, so this is actually rather comparable to what you talked about, Blessing. So we went out and we collected graphs from the standing repetitions of the clone trials, and then unfortunately the graphs took really poorly. So we were left with 10 out of the 50 clones, ranging from 0 to 40% rot. We also selected 5 fungal isolates from our from our colleagues in-house that's working on the fungal genetics. And we then made a matrix consisting of 50 interactions between spruces of various resistance and fungi of various virulence. And for the sake of the transcriptome assay, we also made wounding control. We harvested this at different time points, 5 days and 21 days, in order to see the progression of things, if there was any. Downwards, basically summarized. And as you can see, there's a bit of a mess. And as Andreas said yesterday when he was presenting poster 16, if we actually integrate what we see over all these data, there is no really significant difference. It's integrated over the different isolates. But the actual ranking might differ. And here is then the two isolates Anand was using for his first study. And as you see, the individual ranking is not it's not one-to-one, but it's actually not significantly different when we test it between the two isolates. But if we test it, wow, it works if you point to the screen, that's fantastic. Then both the host term is significant and the virulence term of the pathogen is significant, but the interaction term is not significant. So basically the take-home message from this heat map here is that a good clone is pretty much a good clone. And actually what I've not managed to show here was, because it gets a bit crowded, was the sort of prediction of resistance. This is predicted to be the worst clone. It is the worst clone. This is predicted to be one of the better one. It is the best one. The stuff in the middle, yeah, that's the stuff in the middle. So that worked before, now let's try. Yeah. So we also then wanted to do RNA-seq for our second question, but then from economical purposes, we really didn't have the money to sequence all 50 interactions, which would of course have been interesting, but then we would probably be even further stalled with the analyzers. I'm good. That's good. So we selected 3 clones. So basically now we have selected extremes. So lesion length formation and 3 isolates with also quite distinct differences in lesion length formation on these 3 clones. We sequenced them at 5 days and 21 days, and at the depth where we could do then look at the expression genotype by genotype. There were a number of samples so we could do that. And what we see here is then really that it is the interactions that forms the longer lesions where we have also the most differentially expressed genes. When after the expressed at that time point, and that actually also is true at 5 days post-inoculation. It doesn't really look very different. When we look at what's happening, we have done here a PFAM domain enrichment. We could have done geoterm enrichment. We chose to do a PFAM domain enrichment. We see that the short lesion interactions do something which is different from the clone that does the long lesion interaction, but also in this clone something different is happening in the interactions where we do really long lesions as compared in interactions where we really don't see that long lesions. So if we break the data slightly differently, and we haven't really completed the analysis to the stage we hope to be where we submitted the abstract. So we basically what we see here, and this is the best way I think to visualize what we're seeing. Otherwise there would have been a marathon through heat maps, and that's not really helpful, usually on a big screen. What we see here is that actually these genes which are differentially expressed, there is not so much of a clone specific expression pattern. It's more of a it's more of a sort of interaction-specific expression pattern in each clone, with the exception here of this very vulnerable clone, where we do seem to have a large overlap, and that overlap is clearly defense responses. Where we have the overlaps we have between different clones here, we can see that when we start to look at who's there, then it's starting to make a bit of a sense. So here we basically only have 2 genes overlapping between these 2 interactions in this clone. One of those 2 genes is this probable RLK, which has a very distinct expression pattern. In the 2 more resistant clones, it's expressed clearly with 2 out of 3 isolates, but it's not expressed with the 3rd, and it's either not present or not expressed in the vulnerable clone. I wouldn't know at this point, but this is something we're going to follow up on. So, we are at the level where we can say that in a uniform environment, then the resistance levels we score in an individual genotype or in a genotype of spruce isn't really necessarily species or isolate dependent, but as soon as we shift the environment, things happen. Things start to look very, very different, and that's what Andreas is showing on poster 16. So this is something that we will have to contend with in the breeding program when we devise how to test. But the flip side of this study is that the artificial inoculation method that we work with, and that is worked with in Göttingen as well and other places, it is giving some kind of approximation of what's going on in the field. And that's because that's... And from this follows that a good clone, however we test it, is likely to be a good clone, and a bad clone, however we test that bad clone, is likely to be bad or genotype, which I think is very encouraging. Actually, everything else would have been really horrid to end up with. And we have identified some candidate genes which we think are potentially associated with expression of resistance in the more resistant clones, which we will follow up on, but I will have to come back and tell you more about those at a different conference. The work is, of course, not to a very great deal mine. It's mostly Anand's work. This was his PhD work. And we did that together with Janne Stenlid, who is retiring next month. This feels really awkward. He's been around for all my career, and now they were doing similar work, are doing similar work on the fungal side, which I have not presented today. And Skogforsk is always our very important collaborator because they coordinate all the materials in Sweden, and they were very gratefully felling trees in their clone trial so we could generate grafts. So, and then of course the funders needs to be acknowledged. Thank you.
Thank you, Malin. Thanks. So the next speaker is Yang-Gil Kim from the Seoul National University. Here is
Good morning, everyone. My name is Yang-Gil Kim from Seoul National University, Republic of Korea. It was really long trip to be here. So finally we met. It's really nice to see you guys. And my research is about pine wood nematode, which makes me big problem in South Korea and Northeast Asia, and also in here, Portugal and Spain. So here's our study about to find candidate gene to using transcriptome analysis. So I think most of you already know about the pine wood nematode, but here's brief information for you. Pine wood nematode is parasite nematode of pine trees and cause pine wilt disease. If pine tree get pine wilt disease, they will killed about 6 months later. Is monogamous genus. So when pine tree died by pine wilt disease, the vector will come and they lay eggs into the wood of dead tree. And egg became larva, and larva became pupa, and then nematode will come to pupa. So when pupa became adult, and adult will carry nematode to other pine trees. So it goes on, and one dead tree became 100 and 1,000 to million dead pine trees. So it goes on, problems in Northeast Asia. So actually, pine wood nematode comes from North America, and the pine trees in North America has resistance to nematode. So there is no problem in hometown. But about 100 years ago, pine wood nematode comes to Japan first. And pine wood nematode comes to South Korea, China, and Taiwan. So now it became a serious pest in Northeast Asia. And also it comes to Europe in 1999 in Portugal, and about 10 years later it comes to Spain. So in Korea, in 1988, the nematode is first reported in this city, Busan, which near to Japan. So we assume that this nematode comes from Japan. And before, until 2005, they only distributed in southern part of Korea, southern part of South Korea, because they like the warm temperature. And this monogamous alternatus is the only vector insect at that time, and they move nematode to Pinus densiflora and Pinus umbergi, 2 species. But from 2006, nematode comes to north. They are moved by another vector, monogamous Salterius, and there comes a new host, this Pinus coraensis, which distributed in those parts of the Korea. So now every area of South Korea has been distributed by pine wood nematode. 100,000 it is and also nice pine 2008 in Spain. So this is the Pinus sylvestri is also susceptible to this nematode, but luckily I heard that there's no natural infection has occurred because they distributed in the cold area. And this Pinus pine and Pinus halebusis is also has resistance to this nematode, so they're okay. And there's a one vector species which carry nematode to pine in Europe, monogamous Galloprovinalensis. And this is only vector that I know. So this is our goal of research. We think, as I mentioned, there is we want to find out maybe there will be some genetic difference between two species. So we select these susceptible species. This inheritance so last year, 2021, we 20 trees per each species into experimental forest near Seoul. And then we inoculate pine nematode in June. So, and we at the time we inoculate about 10,000 nematodes into experimental group of 9 trees, and for control group we only do distilled water, and then we collect the cambium samples for just before inoculation. I will call it just 0 week, and then after 2 weeks and 4 weeks after inoculation. So we got three times. Berman funnel method to find out whether they have pine nematode and died. This is too complicated. I just summary. For transcriptome analysis, we extracted RNA using kit and then make it to cDNA library, and then RNA sequencing and trimming and refining and read mapping, and finally genome mutation. Okay, here's the result. So we after 6 months, 5 out of 9 inoculated Pinus densiflora, which is susceptible, were impacted and killed by pine nematodes. So we choose 5 trees, and in contrast, every 9 inoculated Pinus strobus were alive, so without pine nematodes. So this experiment is going well, I think. And so we did RNA sequencing for at least about 4 gigabase pairs to 7. And then we get de novo assembly, because there's no reference genome in Pinus strobus and Pinus densiflora. After refining, we got genes for each transcriptome. Here's the PCA analysis for the flora susceptible, and this right one means the Pinus strobus, which is resistance. As you can see, this red dot is 0 week, so before inoculation, and this triangle green one is for 2 weeks after inoculation, plus 1 is 4 weeks after inoculation. So you can see for susceptible species, you can see the difference between the before and after inoculations. So 2 weeks and 4 weeks looks similar to each other. In contrast, for resistance species, of course, this 0 week looks different from others, but also you can see there's a big differences between 2 weeks and 4 weeks. So the analysis, one heat map, you can see this grouping, weeks and 2 weeks, and this 0 weeks. This is the susceptible one, Pinus densiflora. You can see the difference grouping of between before. In contrast, interestingly, this for resistible species, this 0 week and 4 weeks became one group, then 2 weeks. So we think there must be some special thing in these 2 weeks. So we can say that for susceptible one, the changes between after 0 week and 2 weeks, and then there is no big difference between 2 week and 4 week. Just sorry, but in this case, something different. Maybe there must be some kind of a big happens in 2 weeks. So we find out that there's about 162 upregulated DEG in 2 weeks of Pinus spruce. So we found that 13 DEGs for no expression in Pinus densiflora. So there must be no genes or no expressions in susceptible one, and 2 DEGs were downregulated in Pinus densiflora 2 weeks. So these are the list. And Harpo find the dialect related to nematodes. So for that we need to study more. Okay, so for the time limits, studies. Yes. Okay. Thank you very much for listening. Thank you.
Thank you very much for telling us about your work.
All right. So, good morning. Talking about a project in Loud Valley Pine, and this is the I guess everybody it's caused by the biotrophic fungus Cronartium porcum, and one of the main characteristics is that it causes galls in stems and branches, and if it affects early on in the seedlings, it can cause high mortality. This has been a very big problem in the southeastern United States because pine is one of the main products or tree crops in the area. And around the 60s and 70s, there was a lot of incidents of fusiform rust, and all that led the industry to get together with the government and create the Resistance Screening Center in North Carolina. And this screening center is mostly focused on screening for rust and pitch canker, but it's also including also other species as well. This is more or less the distribution of fusiform rust across the United States, that is also coincides with the geographical distribution of loblolly and slash pine. And we can see here that there are several areas that are of higher incidence of rust. There's others where the incidence is lower. So there is quite a big variation, and it also depends on what is on the tree, also on the variation of the host in that area. If you're not familiar with the life cycle, it's pretty complex. You can see that it has produces 5 different types of spores. It alternates across the 2 hosts. It passes on to oak, where it's actually the primary phase. So it produces its iridinospores and then teliospores, and then the basidiospores infect the pine, and that produces the galls. And after about 6 months or so, that you can see the galls. And late in the fall, it produces these droplets that contain pycnospores. And then in the spring, we can see one of the main characteristics of the fungus is the production of achiospores in the spring, and it's very, you can see them out in the field pretty well. Now we have the, let's say the advantage, or it is quite a good thing, at least for pine, that pine is considered the alternate host. So the rust doesn't pass on from pine to pine, but rather has to go through oak and then again to reinfect pine, which is an advantage for breeding, as it has to go, it is harder to overcome that resistance. As I mentioned before, the pine is the alternate crop, and there's no pine-to-pine transmission. So that is one of the things that we are using to our advantage to look at, to study the effects of pine and of fusiform rust in pine, and to look at the breeding, to look for breeding in this pathogen. So one of the things, one of the characteristics of fusiform rust is that the qualitative resistance is a gene for gene type of interaction. So if we have the fungus, it enters the cells and produces these avirulence proteins that get transferred into the host cell, and then if it encounters a complementary or a reactive resistance protein, then it will trigger defense, which will result in no galls. If they're not compatible, then it will produce a gall. And we can see here more or less the effects. FR1 is a fusiform rust resistance gene that has been that was discovered at least through different through studies with where the host is is heterozygous for FR1, which is a resistance gene. And there is also the Rust genotype that has the AVR1 gene, so they interact, producing resistance. This was a work of Tom Kubitschek from the U.S. Forest Service in the 90s. And so what we see is that if we infect pine with with these spores, whatever the ones that will be interacting with the resistance gene will be resistant, so they won't produce a gall. But the ones that produce a gall, then we can collect the pycnia, and all these pycnia will be of the AVR1 genotype. So we can sort of filter out, so to speak, the ones, those AVR genes that are interacting with the resistance gene in the host. Now, what happens, because of this is a gene-for-gene interaction, what happens if that resistance is in fact overcome? Or, looking ahead. But there is still hope. We have also observed that there are differences among those galled families that are susceptible. There's a variation in the susceptibility in the form of the gall size. So, and this is it's a quantitative caused by the interaction of multiple genes. So these were observed in and we can see that, for example, here these are clones that were tested with fusiform rust, and there are certain families that are more resistant in terms of the score. That means that 0 is totally resistant and 1 is galled. So it's a binary trait, and then these are averages across families and clones. So there are some that have smaller galls, which can be used to our advantage to achieve more durable resistance. So this is kind of the new project that we've been working on for the past year. We're trying to look at it on both sides of the host and pathogen. So we have our first aim is to look at the genotypes in the host and do whole genome sequencing in the pathogen to look at resistance gene and to characterize SNPs that are significant for resistance. We also have a very wide range of different trials that have been carried on historically at the university and through cooperatives. So we have populations, for example, this population, the C-clones population is a family, is the one that I showed earlier. That's 70 different families mated in a circular mating design. This ADAPT2 population is an association population, so they're assumed unrelated, so it was used for GWAS analysis. And they have also been used in trials for fusiform rust resistance. So we have pedigree information, we have phenotypic information, we have genotypic information, and this will be used to do an integrated model that integrates both host and pathogen to look at major gene and quantitative resistance. And finally, we will do a large-scale inoculation experiment to validate this model using an expanded host pathogen matrix. So our first step is to develop markers in the host, and using a study, a first inoculation experiment, where we use 5 different loblolly pine elite families. This includes also one family that we know has the FR1 gene. It's heterozygous for the gene and for that gene, and we have 10 CQF that's Cronartium isolates. So those were obtained from different areas throughout the geographic region, geographic distribution, and constitute of a spore mix of each of those regions. We have the phenotypic data on gall presence and absence, that's a qualitative trait, and we have the quantitative trait of gall length. And then we have the molecular data where we are currently doing the SNP identification and sequence capture for genotyping the Pinus genome of these 5 families. And we're also starting the whole genome sequencing from the pathogen. So last summer, we measured the lesion, not lesion lengths, but the gall lengths. And then in November and December, we collected individual pycnia drops. We're assuming that each pycnia drop they're haploid and they are all the same genotype, but we'll find out if that holds with our sequencing data. And then we will also be sequencing the basidiospores that were used to inoculate these pines. So we will have the genotypic information and the whole genome sequence information of the spores that were used to infect the pines and whatever filtered through from in the pycnia. So this is more or less the methodology. We can see here the different areas. So each colored area is a zone. So each isolate mix was obtained from each of these different zones. And there was a couple of them that were combined too, because these are like 12 zones, I think, and so we had a total of 10. The spores were collected, and that's something that the Resistance Screening Center does every 5 years or so. They go throughout across country, and they collect spores, and then they germinate oak seedlings and infect these. Yes? Oh, 2 minutes, okay. So I'll hurry up then. And then they do the screening. So for results, we saw that there are differences across families in the presence and absence of galls, and that genotype and that response. So at family 5, we know it's heterozygous for the FR1 gene, and we can see that some of the families are pretty resistant, but they also have some susceptibility to each of these spores from each of these areas. We also know from the gall length data that there are also quantitative responses, and there are significant responses across families as well. So we know that, for example, this family this spore combination had produced shorter lesion lengths, but also there are families that have shorter or larger lesion lengths depending on the family. This is the current status of the project at present. We've done most of the collection and most of the genotyping and sequencing is underway. And the next step is now to integrate the model from the incidence and severity from these data, along with historical data, to look at, and along with genotypic and pedigree information, and do predictions based on major gene and polygenic effects. And finally, for our model validation, we'll be doing next spring or summer these crosses, inoculations between the isolates and families, and we are on the lookout currently for rust in the field. So if you know of anybody in southeastern United States that works with pine, we will be very appreciative of them doing some collecting, and we can send out some little kits to collect the spores that will be used for the next inoculation phase. So to end up, we observe family by isolate interactions that could help predict resistance genes that could be deployed in the landscape, and we can also, that would allow us to kind of tailor a little bit more the deployment of better varieties, or varieties that are more resistant in different geographical areas. We also hope to provide a better understanding on the quantitative nature of resistance, and in the big picture view, we hope that this would also provide a bit, be a base that could provide a lot of information across the long life cycles. And with this, I end up the presentation, and we'll be looking forward to your questions.
Thank you. Thanks. So now we have a discussion. I'd like all of the speakers to come to the front. So Tanya, you can stay here. Maybe just put the pointer down. Blessing, Ron, and Young. So I will hand the microphone to one of you. We have two microphones. Anyone would like to start off with a question?
Okay, yes. I can warm up with a sort of a straightforward question to Blessing. Did you measure water potential or try to quantify drought stress in your plants?
No, we just measured the water content in the soil.
Okay, because water potential measurement is pretty easy to do, and I mean you have to get up in the middle of the night, but it's sort of fun, and it's very robust, I think. So.
Good idea for next study. Thank you.
You can also measure it midday.
Yeah. Okay.
Okay, we go.
This question is for Malin, or actually a couple of them. I was curious how you estimate relative resistance in the field. Do you look at stumps? So you cut down, okay. And thinking about what Tanya was talking about, you use different isolates of the fungus, right, to test the different clones. What about using mixtures of isolates for individual inoculations? So that sort of maximizes your chance of hitting them with the worst or whatever you, and so simplifying your assay. And I was wondering also, all the numbers of genes you were showing, how much do you think that is related to defense versus a consequence of the interaction?
Okay. There was a number of questions there. If we start with mixed isolates, yeah, maybe that's a good idea. I never tried. The reason we are using these two tester strains is that we in general in the lab then have a feeling whether we flunked the experiment or whether it's really working. So that's been the reason for us using pure isolates. I think Eva is one of your supervisors is sometimes using mixed isolates. And I don't know how that has been working for her, but I know people are trying. I'm just a control freak. for the question, what is a consequence of the interaction and what's related to sort of controlling the interaction, I don't know at this point in time. I would need to, I mean, we have a number of unsequenced interactions. So our original plan was to do this sequencing and then go back into the other interactions and follow up. But then there was a pandemic and not much happened in the lab.
Just as a quick follow-up, the reason I asked is because if I did not misunderstand you, it seemed like there were many more genes involved in the susceptible interaction. And also, you looked at these gene expression analyses kind of late following the inoculation, right? Because it was up to 21 days.
Yeah, but the pattern in the 5 days is not essentially different. When you have a long lesion, you have more activation of defense responses. And I think that is, if I may speculate, and that's really pure speculation now, that to control heteropathogen, I think it was you that was talking about the formation of lignous tuberized zones and boundary zones around the in the progressing pathogen. And I think what happens, and I think Alejandro Soya had shown that previously, is that when you have a successful interaction with the pathogen, then the pathogen will break through these boundary zones. So in the shorter lesions, I think, but I have not proven it, because I haven't had a chance to test it, that we have a more successful boundary zone formation, and basically in the long lesion settings, that's lost, and they keep trying with what they have, and this is why we see this large induction of genes and sort of broad defense responses. Would you agree?
Yeah, it's for Malin again, and you may keep the microphone as well. So for your, again, gene expression studies and RNA-seq, did you see any repeated patterns between the isolates that you tested on various planes, meaning whether you see the similar responses or whether you could have a common suspects across the planes?
So basically, I was showing this one gene. There was, and the thing was, the intercept across the species, well, within the species, first of all, was very small, and across the species was relatively small. Also, no, not the species, across the isolates, genotypes, sorry, I'm getting mixed up. I haven't had my morning coffee. Or not enough anyway. So the overlaps are surprisingly small. I had anticipated larger overlaps. That's basically our conclusion from this, and whether that's just specific to this experiment or whether that's actually something that we would see, I don't know. But I would say there are relatively few hot candidates to work across all the 3 clones. And that's the only thing I can say at this point.
This is to Tanya. In your phenotypic measurement for quantitative resistance, when you look at gall length, I don't know if this is something you are yet to do, or this is something you've already done, and probably I just missed the slide where you showed probably gall length for quantitative resistance. But my question is, did you also consider maybe the height of the plant itself relative to the length of the gall? Because in our case, when we looked at the galls, from Acronatus magnesii, we do consider not just the diameter of the gall, but also the diameter of the stem, because if you have plants that grow really fast, even though they are susceptible, they tend to produce much larger galls relative to plants that are probably maybe much resistant, also growing very fast. You see some differences, so I don't know if you consider that as well when you looked at gall length as an index for quantitative resistance.
We did not measure the total height of the seedlings. They were,
I still have one more.
in a way, even though there were different families, they were pretty much all, germinated at the same time and so on at the same time. So they were I wouldn't say like totally uniform, but they were pretty
Much the same height.
much the same height, yes. But we did not take any measurements on total height, so that would be something to consider for the validation experiment as well.
Okay, and then at the family level, do you look at probably the percentage of maybe the plants with the gall? I believe probably not all of them produce gall. Maybe some, like the percentage of which gall produced among the plants within each family, also as maybe a measure of aquatic resistance among families.
Yeah, so so you're asking if we looked at the percentage of gall
Yeah,
families?
percentage of plants that produce galls in each family.
Yes, so we did look at how many of these, of the percentage of individuals per family were galled, and then out of those galled, we took the measurements.
Okay.
I
Yeah.
think we'll move on. You can continue
Okay.
during the coffee break, maybe, because there are a few more people. So we have one question way back here. Sorry, I just need to get this microphone's not working. I'm going to take one more after that.
Hello. This was a follow comment with this multiple inoculation. I think, at least in my head, the problem with inoculating more than isolate in Heterobasidium is that these are heterokaryotic isolates, and they will, once you will put them in the lesion, they will start competing between themselves because they will sort of try to sort of lock down each other. So I think this is one of the problems, actually. One could think how this happens in reality. I mean, you have many genotypes arriving into a stem, but then you can see much, a fewer amount of them when you look at the tree. So there's sort of a filtering, and there's like sort of a winning isolate that at the end sort of outcompete the other. So I think in my head it would be better to try to replicate that single isolate attack. That's what normally would happen in the forest, rather than challenging a lesion with different heterokaryotic isolates. But I was wondering a bit, how do you do that with the rust? Because do you inoculate with basidiospores, and then you expect them to sort of fertilize themselves, or how is that?
Are you saying we?
Yes. So basically, how do you visualize this sort of mixture of isolates, or what do you mean with that? It's like a
Basidiospores. We infected with basidiospores, and then with a mix of basidiospores, but for the big inoculation, we will do individual isolates. Is that more or less?
Yeah, that sort of answer. So you don't expect them to sort of compete, or what is your so? How do you expect the pathogen then to react in this sort of inoculation point? So will they create different individuals or?
Can you repeat the question?
Yeah, so if you put a mixture of basidiospores, I understand, from different isolates, and you put them in the lesion, sort of you inoculate them, then they will sort of fertilize between, and they will create like a population of isolates in that. So,
Yes.
So is this what happens in reality, or how do you expect if they will compete, or do you have any thought on that?
Well, we don't know if they compete, different isolates among themselves. It's something that we will probably answer after we do the sequencing, see which ones were filtered out, at least for the susceptible, genotypes. Sorry, I feel it is a little bit.
Okay, one last question from the audience.
It's regarding the nematode work. You inoculated 2 and 4 weeks, the trees. So what happens, it seems like a really long period of time. So what happened in the period between 0 and 2 weeks? Are there any responses, or is there any data to support why you did 2 and 4 weeks?
You mean the phenotypic variation? Actually, we take picture every 2 weeks. Until about 10 weeks, there's no difference. So just there is a pre-study before me. So another researcher in Korea, they inoculated big trees, about 10 or 20 years old. So they also try sample on 2 weeks and 4 weeks. So we want to try the same conditions too. After maybe we can compare the results. So actually there's not firm reason to take 2 weeks and 4 weeks. It's just kind of a conditions.
Okay, thank you. Blessing wanted to add something?
No, I have a small question for him because I'm not used to finding nematodes. How did you inoculate? You said you used water for the control. Did you inject the food or how did you do it?
Actually, I saw the picture of your picture, right? You show the picture to cut and inject, right? It's similar. Just in my lab, we grow. We make medium for nematode, and then we kind of collect my nematode to water, and then using the pipette, we cut the cambium of trees, and then they put some cottons, and then we pipette to about 100 microliter water, and then after then we just lapping. That's all. Yeah.
Okay, thank you. I had one last question for Tanya, if your voice is up to it. You described resistance as a gene-for-gene mechanism, and you described the formation of the galls as kind of a presence or absence trait. But in the light of the present work, do you think that's still the best model to describe what's going on at the general population level, or are there other loci, and is it more quantitative than purely qualitative?
So the resistance is mostly it's primarily gene for gene, but there are different studies we've seen. There's at least around 9 resistance genes, and some of them are like about 5 or 6 of them are clustered in linkage group 2 of the pine genome. So that's what we know so far. So there is some variation in the response. We don't know if all the genes, or if there are any additional genes, or if there are any pseudogenes, for example. But that's the primary response. But in the case of long-term resistance, we would like to know which of the current resistance genes if there's any overcoming of that resistance in the population, for example. But we're also looking at some cases where they've deployed different families that were supposedly resistant, but some are showing galls, for example. So there is a possibility that some of the resistance is being overcome.
Okay.
So those are some of the things we're looking at.
Okay, thank you very much. Please join me in thanking the speakers for the presentations. And coffee break now. Thank you.
You are not at the. Let know. Otherwise, it's going to we are going to have the tomorrow for the trip. Great. Speaker is a very we share Catalonia.
Thank you. Nice. It was a pleasure. Well, to the concept of adaptive syndromes in forestries, which I think is quite related to the object of this workshop about tree-parasite interactions, because at the end the genetics of the tree-parasite interactions, it's in close connection with the shaping of the adaptive syndrome in a forestry species. So I'm going to briefly provide you an we have been up trials can provide. So this is the outline of the talk. I will start with a brief introduction on the topic. Then I will try to contextualize the concept of adaptive syndrome in the general frame of this workshop. I will later on talk about general strategies and some approaches that, as I said before, we have been applied so far during the last times. And finally, I will end up with challenges and conclusions. So first of all, I think it's informative to provide a formal definition of an adaptive syndrome. I think that many of you are familiar with it, but in any case, an adaptive syndrome is the covariation that present a. So when two traits present give rise to what is called an adaptive syndrome. And I'm going to illustrate this with a very simple idea in which you may see here a couple of individuals growing side by side. The left one invests a lot in aerial growth, while the right one invests or allocates more resources to roots. So as you can see, there is covariation between these two functional traits, and well, this would define a part of an adaptive syndrome for this hypothetical species. The point here is whether to see if this situation actually exists in reality, and if we can characterize this situation in the reality as well. So the answer to these two questions will be yes. This situation actually exists. I'm going to provide you with an example for Pinus alepensis, and we have characterized this adaptive syndrome by means of two different approaches: the use of a georadar, which is a geophysical device that allows to see, let's say, below ground the surface, and the usage of, in parallel, of stable isotopes in xylem water. And both approaches give about the same results. I'm going to summarize them in this sketch. I'm going to compare three hypothetical individuals of three populations from Aleppo pine. Another individual from southeastern Spain to both aerial growth and roots. The individual from Spain will show what is known as allometric scaling, but if we go to the eastern part of the Mediterranean, a typical archetypical individual from Greece would show this pattern, which shows a preferential allocation to aerial growth rather to a deep rooting. So this will define a simple adaptive syndrome for this pine species. So all we agree, I think, that a current big issue in forest genetics is to understand and forecast, for sure, the responses of our forests to the environmental challenges posed by global change. But this is particularly important to do this from an evolutionary perspective, because this may give us clues about how to deploy more efficiently forest genetic material and how to delineate better assisted migration strategies, for instance. And, well, a primary question here is to understand how life history traits interact with each other in order to give rise to also understand the adaptive syndrome of global climate change. I think it's important to understand those traits related to tolerance to abiotic and biotic stressors. All these functional traits related to these stressors would eventually define the adaptive syndrome of a particular forestry species. And what I'm going to do is to present some results using provenance trials as a preeminent tool. Provenance trials have a long tradition in forest research, dating back to at least 100 years ago. So they are super useful in order to separate genetic and environmental effects, but they have problems, of course. Sometimes they are poorly maintained and kept, and I'm going to show you here a couple of examples. We were surveying a couple of trials in Sardinia last autumn. This will be a super nice Pinus pinaster provenance trial from Vallehermosa in Sardinia. And 30 kilometers south, we had another provenance trials, in this case from Aleppo pine, which is almost like a jungle. You may hint in the background some of the remaining black Aleppo pine trees. So, yes, we have to cope with this, but altogether I think these provenance trials are super informative. So I'm going to talk about adaptive syndromes, and I'm going to try to contextualize the concept of these adaptive syndromes by using an example of Pinus pinaster in relation to the I will show you some current approaches that we have been applied to the case of Aleppo pine. So, starting with the contextualization. So, as I said before, we're going to use provenance as adaptive units in forest trees. And I'm going to present here an example of 15 trials of Pinus pinaster. Here you can see the triangles, in which more than 100 populations had been evaluated, which are the dots. The different colors represent different genetic groups or meta-populations of this Pinus pinaster. And I'm going to represent here the functional variability of the species, summarizing this functional variability for 3 height and for these 8 genetic groups. This functional variability is really huge. You may see here how populations from Northern Africa grow really not too much, about 8 meters at age 20, while populations from this area, from Atlantic, Galicia, Portugal, and France, may grow up to 11 meters. So this is just the tip of the iceberg of the functional variability for this species, but of course there is much more behind. And I'm going to try to relate these results with the resistance to a particular pest that affects this species, which is the maritime pine vast scale Machucoccus feitoidi, which is an endemic species, an endemic pest of the Iberian Peninsula, but which has threatened a lot the populations of maritime pine in Italy and France. But incidentally, it happens that this pest has become very damaging also in Spain, and this is a press note indicating, like 5 years ago, indicating that there has been a rather large amount of money devoted to control maritime pine dieback in northeastern Spain. And as a predisposing factors for the incidence of this pest are the recurrent droughts and the insufficient management. This is what the forest technicians think about why this endemic pest has become really damaging now in Spain. But on top of these predisposing factors, there is the genetic factor, which the technicians themselves call the extreme genetic complexity of the species. Genetic variability at stand level, imported seed from Liguria in Italy. This imported seed is very sensitive to Maxococcus. So in order to put some order in this conundrum, what I had the chance a few years ago to analyze a multi-environment trial of Pinus pinaster, which consists of 4 trials in Italy, where 8 populations had been evaluated, 5 from Spain, 1 from Morocco, 1 from North Italy, and another for Corsica. And it happened that about 10 years ago, one of these trials, particularly the Bordighera trial in Liguria, was heavily affected by Maxococcus in a way that most of the trees died, except few trees, most of them coming from the Moroccan population. And by putting together all the data of this multi-environment trial, which is summarized in this simple figure, I think that this simple figure is super informative on part of the adaptive syndrome of this species. And for instance, we have here in the x-axis the growth potential of the 8 populations, or 7 in this case, and in the y-axis the pest incidence in the Bordighera trial. And you can see the relationship is almost linear. And we had here the Corsica and the Italian populations characterized by a very high growth potential coupled with a very high sensitivity to Maxococcus. In between, we have the Spanish populations, and finally, we have the population from Morocco, which has both low growth potential and high tolerance to Maxococcus. This defines a typical trade-off between these 2 traits, which is part of the adaptive syndrome of this species. So, in order to characterize the adaptive syndrome of a particular species, we may think about as a general strategy, which I'm going to show here, which is super general and probably, it adds not too much, but anyway, we have essentially today 2 types of broad methods, genotyping and phenotyping, both tending to be high throughput, if possible. That's it, cheap and cost effective. In the case of genotyping, it's clear. In the case of phenotyping, it depends. And as materials, we have genetic trials and sometimes conservation units and germ plus banks. So these methods can be applied straight to our genetic trials, say provenance trials. And these conservation units and banks may act as a sort of backup in order to restore or renew these genetic trials. And by applying these methods, we get a corpus of data that needs to be properly analyzed, of course. So, based on this general strategy, what are the available approaches to forest geneticists? So I have distinguished, sorry, too fast. I have distinguished 3 big types of approaches. One related to phenotyping, another to genomics, to the combination of genomics and phenomics, and finally the possibility to model all this body of information. Phenotyping, what do we have at our hands? We have the possibility to apply high throughput phenotyping techniques using images, usually also obtained from drones, which I will refer later. We can gather information on ecophysiology and phenology. We have alternative traits that can be useful for defining the adaptive syndrome of a species. These alternative traits are probably not mainstream ones, but they can be really useful. I'm talking about, for instance, the investment in reserves and, of course, in defenses, the distribution of roots, water use efficiency, soil water extraction patterns, etc. And finally, I have included here a 3rd type of phenotyping techniques, which are also drone-based, which allow to characterize the morphological variability of the trees. I'm talking about essentially LiDAR techniques and RGB data. Also, we can combine genomics and phenomics. I will go later on to this. And finally, we can use different types of modelings. I am mentioning here, for instance, structural equation modeling or GVI analysis. These are two of the many array of strategies that can be applied here. For issues here on topics. So for this purpose, I'm using as a study case a Pinus halepensis provenance trial, which is located in eastern Spain, in which around 60, almost 60 Mediterranean hog populations, as you can see here, the dots, have been tested over the last 30 years. This trial, I can tell you, is a joy to work with because it's super regular, it's super uniform, which means that, well, most of the variability that you can measure here is genetic. It's a genetic one. I did nothing. So, first, we can apply, for instance, a high throughput phenotyping via the use of drones and different types of cameras, RGB, multispectral, or thermal. So what we did was to do several flights in summer, autumn and spring, following this path, using this particular drone, well, doesn't matter. And what we did was to obtain this sort of imagery that you can see here. And essentially, what these cameras allow is to retrieve information on the visible and near-infrared spectrum of the light. And by combining different wavelengths, we can derive different vegetation indices, the very well-known NDVI for sure, the photosynthetic reflectance index, one index related to photosynthetic pigments, another to the amount of water in the needles, for instance. This is just an example. So what we did is to bring all this information to statistics. So we ran ANOVA. This is super easy. And what we found here, for instance, is a lot of population differentiation in some indices related to leaf area, which would be these ones for summer, autumn, and spring, to chlorophyll content, to different photosynthetic pigments, also to water content in needles in spring, and finally to the amount of transpiration by each individual in summer. I'm going to use this information later on. So that's it for now. Another possibility is to use phenotyping techniques for the morphological characterization of the trees. Basically, it consists of combining RGB and LiDAR information. This is a twin trial to the one that I showed you before. It has the same populations. And what we did is to perform first a crown segmentation analysis. And based on this individual crown segmentation, we were able to identify each individual tree. And imposing this information on LiDAR data, we got a cloud of points for each of the about 1,000 individuals of the trial. And well, this is very, very nice because all these clouds of points allow to infer, in this case, 23 LiDAR indices, which are related to, for instance, crown architecture. And they show in crown volume, in the ratio between crown volume and crown area, and in the height of widest crown section. So, another big types or great type or large types of approaches has to do with the combination of genomics and phenomics. And on the left-hand side, you have the same type of data that I told you before for that Aleppo pine trial, which means that we have vegetation indices for a number of individual phenotypes present in that trial. And on the right, we have genotypic information, typical one, SNPs, etc., and we can perform genotype-phenotype associations. This is a typical GWAS analysis, and these are the results which tell us that, of course, we may identify some particular markers behind some meaningful ecophysiological traits like leaf area, photosynthetic pigments, water content, canopy temperature and growth trace. And eventually, some of these SNPs are related to known functions of the gene behind the marker. And this is an example I'm going to go on. Finally, I'm going to talk briefly about modeling of this corpus of data, of all this phenotypic data, and I will provide you an example about structural equation modeling. You may be familiar with the technique. This is widely used in ecology, but it's not so used in evolutionary genetics, for instance. And this SEM, what it does is to estimate complex processes that cannot be directly made these processes through observed variables, some variables that are physically measured, which underlie the latent variables. So what we did was to apply SEM to analyze the relations between functional traits and the adaptive structure of a species. I think I'm fine with, no, it's fine? Okay, it's 3 minutes, I think. So what I did, or what we did in this case, is to use SEM in order to disentangle the adaptive structure of this species. The model that we use is this one. So we had on the left 3 latent variables related to resource acquisition. For instance, water uptake depth by each individual, transpiration and phenology. These are related to resource acquisition, either water or carbon. And we related these source variables to sink variables related to reproduction, reserves, or growth. In other words, what the individual can do with these resources that are acquired. They can be devoted to reproduction, reserves, or growth. And these variables were not measured, but instead we measured some surrogates of them. And for instance, in order to know what about how deep the tree goes in order to take up water, you use stable isotopes. In xylem, we use multispectral data for phenology and transpiration. We use counting for reproduction, amount of sugars and starch from reserves, and primary and secondary growth for growth. And this is the final model, and essentially what it tells is that for Aleppo pine, those populations which are able to support transpiration during summer, in turn, are capable of growing more. Also, those populations that are able to take water from the deep soil are reproduce less, comparatively less. The populations that are able to photosynthesize a lot in spring accumulate more reserves, but they reproduce less. And also related to the sink variables, there are a number of trade-offs among them. Populations reproducing a lot grow less, and populations that grow a lot, they accumulate less reserves. And this is the value of this relationship. So altogether, this gives us information on the adaptive syndrome of this species, which can be summarized in this slide. So we know that this was how the post-glacial recolonization of this species happened, by moving the individuals from east to west of the Mediterranean, following a north or a south, perhaps, path. And if we focus on this population from the drier extreme of the ecological range of this species, we now know that they experience less above ground growth, they have higher reproductive effort, they have enhanced water use efficiency, they are less dependent on topsoil water, they have deeper roots, they invest more in storage, and they have more symmetric and compacted crown compared with their northern and eastern counterparts or populations. So, and this helps to understand that these species have a very strong geographical structure. There are populations that they are locally adapted to drought, others are adapted to competition for light. There are clear evolutionary trade-offs between aerial growth and others, as I mentioned before. And there is a gap here that the colleagues from Emisión Biológica are trying to disentangle, which is the role of defense in this adaptive syndrome of the species. So, to conclude, I think that we need to be humble and recognize that we still have a very long path towards a complete understanding of the evolutionary trade-offs that shape the intraspecific variability of our forestry species. But on the other hand, it's urgent to know the range of existing intraspecific adaptive variation and the limits to phenotypic plasticity. In order to what? To be able to better deploy our genetic material for reforestation or ecological restoration, for instance, or in order to delineate good strategies for assisted migration. Phenotyping is the actual bottleneck to face this challenge. We need updated tools for phenotyping, new solutions which should allow facing this challenge. And this is a challenge that we have, and this is just to conclude. When preparing this talk, I came across this nice training school, which will be held, I think, will be held next week in Valladolid, or in a couple of weeks, about new detection and diagnosis methods for pests and pathogens, which it's an example of those activities that should help to close or narrow the gap between the phenotyping needs and the urgent need to know about the range of adaptive variation in our species. So that's it. Thank you very much. And well, if you have any questions, I will try.
Thank you. You are in time.
Garden with the drones and so at least they look like well placed. How does those techniques work with real forests where you have different ages of trees, some are covering the others and all that stuff?
First, of course, I would question. I'm not a specialist in drone and on the processing of images. Of course, when we are using this technique as a phenotyping tool in, say, more or less uniform genetic trials, things become quite simple. And for that purpose, I think the technique works very well. Of course, if you go beyond this experimental setup of using genetic trials, things become much more complicated. And even within the range of provenance trials that we can work with, there are some of them that are super homogeneous and uniform, and another ones that are impossible to use for the purpose of high throughput phenotyping through drones. Because the amount of experimental noise that you have there makes impossible to detect population difference or individual difference at the genetic level. So it's like this. So we are constrained by this problem, but at the end, I think the technique is very much promising if the genetic trials are more or less reasonably homogeneous, I would say. So, in any case, I mean, all the techniques such as LiDAR are very widely used as an inventory tool for forest management, for instance, forest inventory. So, yes, but it's another different objectives and other different ideas behind it.
I have a quick question. Hi, I'm Jeff Williams from the U.S. Forest Service. I have a quick question about the trade-off between growth and resistance to the Matsukakus scale, the figure that you showed. I believe it was the Italian populations that were the fastest growing and least resistant to the scale. Right?
Yeah,
so, pardon me?
Can you repeat this?
Oh, sorry. Yeah, the question is about the trade-off between growth and resistance. And I believe in the figure you were showing that the Italian populations and provenances were the least resistant and the fastest growing.
Yeah.
And I was just curious, is that pest or similar pests present in Italy? And if so, like what's the severity of the impact there? Or
so the pests have never been present in France or in Italy until the last 30 years approximately, which means that those populations for sure didn't have to cope before with that particular pest, which suggests that they could be totally susceptible to the pest because they haven't developed any way of defending themselves against it. So this is the starting point. In fact, these populations from France, Italy are known to be like very high. They are known to have a very large growth among the range of the species, and this is one of the reasons that they have been traditionally used for reforestation in northeastern Spain. But this usage doesn't pay off because of the impact of climate change, recurrent droughts, and also the insufficient management of the reforestations that the landowners do in their lands.
Since you said that, I just ask one more follow-up question.
Yeah, sure.
So what do you think the consequences of that type of selection for plus trees or elite growth are having currently for pest outbreaks worldwide?
$1 million question, yeah, sure. Yeah, I think that this trade-off, I cannot say that they are universal, but they are very widely spread all over many forestry species. And my view is that we have to be super careful, and we need to get to a compromise between growth potential and potential drawbacks related to a poor investment in defenses and, for instance, intolerance to drought or to other abiotic stresses. So the trade-offs are almost universally there, and yes, they pose an extra difficulty for breeders, and this is well known.
Which leads me to my question, because I was thinking that it's such a pity that you did not have induced defenses or defenses in general in your structural equation modeling. Because I'm just wondering what relationships you would have found. My guess would have been a negative relationship between growth and defense.
I guess so.
But it would have been interesting to see the coefficients.
I guess so.
How can you not think about induced defense?
So, well, yeah, given the audience, I must apologize for sure. Actually we thought, and the colleagues from Emisión Biológica phenotype, you remember, Rafa and Luis, you were phenotyping that trial. So I'm not saying that it's their fault. No, because But yeah, this is definitely something pending to do, Rafa and Luis, and it wouldn't be too difficult to rerun the models incorporating that information. And I guess it will be like you are saying. Yes.
I think structural equation modeling has so much power to understand systems.
Yes.
It's very cool.
Yes, there is a problem with them that you usually use this modeling with structural equations in order to model linear relationships. Well, sometimes you know that the relations are not strictly linear, but this is another thing.
Some other question?
And getting back to that, maybe there are other approaches for modeling that they're becoming more and more used right now that are specifically for nonlinear relationship, like generalized addictive models. I think would be a good way of trying, so that you could model better.
Yeah, of course. The point is that by using this kind of integrative modeling, you can more or less get a more general picture of the whole, in this case, adaptive syndrome behind the species. Usually for all these alternative approaches, you have a response variable and many independent variables behind, but here it's kind of integrative. And, I mean, it's going deeper into this modeling, this structural equation modeling. I know that there are ways to integrate nonlinear relations. Yeah, but this is part of my scope.
Oh, it's even part from mine, honestly, so I speak for what I've heard.
Yeah, but it's something that should be.
Thank you. Very nice talk, by the way.
Some question? I have anyone?
Jordi, congratulations for the very nice talk. I have a question that is, with a provenance trial, you suppress the environmental effect. Well, my question is if there is interaction between the genotype or and the environment, what conclusions can be extracted from only one provenance trial? I mean, how many provenance trials in different parts should be carried out to obtain robust results or conclusion?
Yeah, this is also a super clever question for sure. Yeah, I mean, when we apply this sort of phenotyping, intensive one in a single trial, then we have the problem that we may be missing a significant part of the whole picture. Why? Because there is usually genotype by environment interactions, which are widespread in forestry species, meaning that the snapshot that you get from a particular trial may not be truly representative of what is happening under different environmental conditions not represented by that particular trial. That one that I use is a very nice trial because first it's super homogeneous, as I told, but also it's representative of the average conditions of the species. So if you, I would go there is a graph in which the average condition of the species are represented in that particular trial. Thank you. Here. So this is the autoecology, temperature against precipitation, and the trial is that red dot, which means it's average. Not too bad, not too good. Of course, for the purpose of phenotyping with precision and accuracy, I would tend to favor trials that are under near optimal conditions, because the amount of noise in the genetic trial used to be much less. But you may be missing what is happening to the populations in real challenging conditions. Although these challenging conditions may differ with regard to the reason of the poor conditions for the trial. Could be, I don't know, nutrient deficiency, could be drought, could be cold, could be, and then the provenances could react differently to these stressors. So, I mean, it's not that simple. And of course, ideally, this exercise should be repeated for at least 4 or 5 extra trials in order to get the complete picture.
Okay, thank you.
Yeah, thank you, Jordi. Wait, I have something in my mind. That is, we hear about the Enrico talk yesterday about your machine learning and high throughput phenotyping with FGIR, with vibrational infrared, and Kate was talking presented something similar. Asier, there on the corner, hyperspect and prediction of terpenes. You are
presenting very nice work about integrating all these things. Okay, defenses, induced defenses remains to be analyzed, but so just wondering that if we can, this definitely is going to be a tool that have to be very common. It's cheap, easy to do, but maybe we more or less, but maybe how can integrate this machine learning that was presented with the F-tier or infrared, near infrared or whatever, the machine learning for predicting the screening, for resistance or screening for anything, how we can use your data with the machine learning stuff. Do you think that?
Well, I'm not too much familiar with machine learning. I guess that you need a quite large population size in order to train the model. Not too much. How many individuals? 500, 1,000, maybe? 1,000?
I guess it depends on the question, but in our case, we were working on screening for resistance against specific pathogens, and you can get by with maybe, I don't know, 70 individuals.
70?
Yeah, something like that.
Well, then, I mean, the possibility is there. I mean, here we have almost 60 populations already, but behind we have like 700 trees. So it's a matter of
All the more power. I mean, the more you have, the better it is.
Yeah. Well, I think that there is a lot of room to apply new techniques like machine learning together with these new phenotyping techniques that are going to be like. For the technician, we are all time thinking on biostatistics, on multiple or genome stuff, but really more mathematical.
Thank you, Jordi. So we are going to. I didn't put the name. We didn't put names to the sessions. Yesterday was more about genetic variation and breeding, and now we are moving a little bit more with this talk about genotype by environment interaction, the effect of the climate or the effect of the local adaptation, and as well to new or to understanding the defensive traits or understanding resistance mechanisms. So now is time, Raúl is going to moderate the next section.
So, thank you. Now we have 4 presentations. After this excellent talk from Jordi Voltas, he opened the door for the environment coming to this meeting, and actually the interaction of the environment and the genetics. Yesterday, Juan Antonio showed that some elm genotypes that are resistant in some environments, they are not in others. So we cannot see the resistance as something fixed, as any other trait is under the G by G interaction, right? So depending on the environments, different genotypes can be the resistant. And before somebody talk about trade-offs, we cannot forget that trade-offs are also context dependent. This is a basic ecological idea. So trade-off also depends of the proper environment. So even the geneticists, we have the environment on our heads the whole time. And we have now 4 presentations that in terms of the title, I believe they are dealing with this question. And so the first one by Soumya Josh, I'm not sure if yeah? So please, we are going to have 15 minutes. Yeah, I'm going to be here.
All right. So good morning, everyone. Late good morning. I'm here to talk about how pine trees become more susceptible to pathogenic attack under climate change. So when we talk about climate change, the immediate thoughts that come to head in terms of forest health or tree health is increased tree mortality or decline in forest health, either by direct or indirect impacts. And then further, this decline in forest health or increase in tree mortality is explained in terms of precipitation and temperatures, where consistent high precipitation and more moderate or optimum temperatures is indicated towards a trend of low mortality, whereas the opposite, where you have severe drought due to lower precipitation and consistently high temperatures, leads to increased mortality of trees. So this was explained in the paper by Allen in 2010, and then with this knowledge, currently we exist in this zone of mortality threshold, where the conditions of temperature and precipitation is sort of sublethal to the trees, and we're gradually seeing more and more increased mortality, increased susceptibility of trees and crop pathosystems alike. So then we usually ask the questions that, is it the same all over the world, or is it the same for all types of pathosystems? And yes, it is. Trees or plants do become more susceptible to pathogenic attack under the influence of climate change, which is explained by drought and high temperatures. However, we do not know how. So in order to investigate that, we chose the Austrian pine Diplodia pinea, Diplodia species pathosystem, mostly because Diplodia pinea is a cosmopolitan pathogen. It is found in many tree species, conifers, dicots, and even some non-tree species. And then it infects the current year shoots, which causes browning and then stunting due to growth inhibition, and usually leads to deformity of the trees and gradual death. It likes water-stressed trees. It's an ectotrophic pathogen. The systemic defenses by the host, which is in this case Austrian pine, is well documented, as well as the defenses that are constitutive, as well as those that are induced in response to the pathogenic infection, has also been documented in the system. So with this information, we set up an experiment in 2016 where we used a split plot design and growth chambers to sort of simulate a climate change treatment or climate change condition. And we also, in a separate growth chamber, maintained a controlled climate condition. And then as a second factor, or the subplot factor, we inoculated the plants either with the more aggressive Diplodia pinea or the less aggressive Diplodia scribiculata, or we just inoculated them with the mock, which was just the growth media. And then the sampling was done at 3 time points, which was 12 hours, 72 hours, or 2 weeks after the chamber are mentioned here, and the results that I'm going to present forward is only from the 72-hour time point. So just visually, we do see, if you look at the picture on the bottom left, the lesions produced by Diplodia scribiculata in the host trees that has already been exposed to climate change treatment was much longer compared to the lesions produced by the mock, which is top left. And then further, when we did an ANOVA for all the lesions, we saw, if you look at the blue bars in the graph, we see that Diplodia pinea on the extreme right produces longer lesions than Diplodia scribiculata under controlled environment. However, under climate change, the lesions produced by both pathogens were similar, which made us speculate what is happening in the host trees due to climate change, which is making the lesions produced in these trees on par with the aggressive pathogen. So in order to investigate that, we looked at the transcriptome, and mostly the differential expression of the host. And we based the differential expression based on 4 comparisons. First, where we compared the Diplodiapenia infected host with the mock induced host under controlled climate. Second comparison, where we looked at Scorbicillata infected hosts compared to the mock under controlled climate. The third comparison, where we looked at the individual pathogen infected hosts with each other. And this comparison, we also looked at the pathogen RNA-seq. And for comparison 4, we looked at what is changing in the host that has been infected with Scorbicillata and exposed to the climate change treatment compared to the control climate. And we looked at the pathogen RNA-seq for this comparison as well. So we ended up with a list of a lot of significant DE genes, which we further annotated using their KEGG IDs and also their gene ontology terms. Mind you, I used only geobiological terms, and only those genes that were annotated both on KEGG and gene ontology, so they did have some sort of function or labels attached to them, but then retained into a quote-unquote most highly enriched and highly expressed genes. And thereby, I looked into them overall, what is the trend, if I can see them through the volcano plots. And if you look at the top 2 volcano plots, where you have the pathogen-infected trees compared with each other, we see there is a much stronger response in Scorbicillata-infected hosts compared to Diplodiapenia-infected hosts, which probably indicates that there's a higher host response due to a Scorbicillata infection. Which can be linked to why we see shorter lesions, under controlled climate. Then when we compared the host infected trees with each other, we see that there is a much more suppression in Diplodiapenia infected hosts compared to Diplodia scorbicillata infected hosts. However, in the pathogen side, we see some very highly upregulated genes in Sapenia compared to Scorbicillata, which might indicate the aggressiveness that the pathogen displays compared to its lesser aggressive sister pathogen. And then under climate change treatment, we see downregulation of both hosts and pathogens, which is indicating that both of them are getting affected. However, we do see certain very highly upregulated genes in the pathogens, which we wanted to investigate further. Then we dig deeper into this, and we categorized our expression in terms of primary and secondary metabolism, mostly based on their relevance to the overall tree health or to looking at primary metabolism. We found that in Diplodiapenia infected trees, photosynthesis was suppressed. We did not see any changes in Diplodia scorbicillata. For nitrogen metabolism, we do see there is an there's an increase in gene expression of nitrogen metabolism genes in both hosts, both pathogen-infected hosts. However, the scale and the number, as you see, is higher in Scorbicillata-infected hosts, and this is even more prominent when we look at the fatty acid biosynthesis genes. There is the scale, the number is much higher in Scorbicillata-infected hosts, whereas we did specialized saw that of defenses in both pathogen-infected hosts. However, as we saw, the scale and the number of differential expression in Scorbicillata-infected hosts is much higher. And then similarly, in the phytohormone transduction pathways as well, we see the same trend, where genes related to some of the critical phytohormones, such as jasmonic acid, abscisic acid, auxin, and ROS signaling are all very highly upregulated in response to a Scorbicillata infection. And thereafter, we looked at what's happening in the hosts infected with Diplodia scorbicillata under climate change as compared to a controlled environment. And we saw, in terms of primary metabolism or carbon, we see severe suppression of photosynthesis, carbon fixation, and genes that are associated with mobility of carbon reserves such as starch and sucrose. When we looked at nitrogen, similar trend, we see fatty acid biosynthesis genes that are downregulated, as well as we saw other genes related to fatty acid metabolism. So mobility of fatty acids, lipids were also getting suppressed. And a similar trend was also observed in nitrogen metabolism. So sort of indicating that due to the climate change or the abiotic stress, the host major carbon, nitrogen, fatty acid pathways are all getting suppressed. We saw that yes, the plant hormone signal transduction pathways were all suppressed, and most of the expression that we saw were all associated mostly with abiotic stress and not very similar to the expression we saw for pathogen response. And the phenylpropanoid pathways and the terpenoid backbone biosynthesis pathways, as well as the monoterpenes, sesquiterpenes, and diterpene synthesis pathways were downregulated, which has been associated with defense in this pathosystem. So overall, we summarize the host and the pathogen gene expression and the various comparisons in this integrated model, where we see that due to a Diplodia scorbicillata infection in the host under a controlled environment, there is enrichment of nitrogen metabolism, there is enrichment of fatty acid biosynthesis, and there is enrichment of defenses, indicating these three pathways are very crucial in the interaction of the host with the Diplodia scorbicillata. However, under climate change, these are the pathways, along with carbon fixation and metabolism pathways, get highly suppressed, which perhaps can explain why the pathogen gains that advantage and is able to spread better, which is sort of my conclusion that under a controlled environment, Diplodia cypinea is somehow either directly or indirectly able to suppress the carbon metabolism of the host. And so even though you have a defense induction, it is not good enough and the pathogen is able to spread. However, under climate change conditions, which sort of takes care of the host's carbon, nitrogen, and fatty acid metabolism and suppresses them, creates a good opportunity for the less aggressive Diplodia scorbicillata to then properly spread, use the sucrose and starch that is already available, and possibly leading to increased susceptibility under climate change. So with that, I would like to acknowledge my advisor, Enrico Bonello, my advisory committee, the Bonello Lab, as well as its past members, and most importantly, the people who gave the money to do the research, so OARDC Seeds and the Department of Plant Pathology. And finally, this was a big paper. There is a lot of information, most of which is not on the slides. So I request, please make an effort, go read the paper. There's so much in this story, so much happening. And if you have any immediate questions from what I talked about today, I'll be waiting for them at the end of the session. Thank you.
Perfect. Soumya, thank you so much for being on time. I believe time for the Peter Orm. Yes. Please, you have your 15 minutes.
2 minutes. Yeah. And just press this one. Okay. Yes, my name is Petter Örn and I come from Skogforsk, Forest Research Institute in Sweden. I'm going to talk about seasonal variation in Norway spruce defense response to inoculation with bark beetle-associated blue-stain fungi, well, in relation to some site conditions. So my main interest is in phenological interactions and how bark beetle interact with spruce and the weather and climate and how this changes over time, both within the season and between seasons. So we have the Norway spruce bud burst, the seasonal phenology, and the the bark beetle phenology and climate risks occurring at different time. So what I have done, I started out doing field work in these kind of areas after an outbreak after a windstorm event in 2005 in Sweden, which blew down 75 million of cubic meters of wood. And this is, historically, this has been the trigger of outbreaks in Sweden in northern conditions. But severe drought stress may also reduce tree defenses, as we have seen more lately. So what I did was using pheromone traps to study flight pattern, and I also had trees infested with bark beetles and put emergence logs into these emergence bags and collecting the beetles. Oh, where is the pointer? Up. Yeah, collecting the beetles here to study spring swarming occurring in May and coming sister brood swarming. That's when they emerge, parent beetles emerge, and also potential second generation establishment. So, and the results of this, the black line shows the pheromone trap catch and shows the most activity occurred in early season before July, actually. And most parental beetles, they emerged to perform a sister brood flight and potentially attack new trees. But this both parental emergence and offspring emergence decreases with thermal sum and during the season. Let me see. Another way to look at this is you could see the logs being infested in spring and having the galleries, forming galleries underneath the bark, and we had the sister brood in June attacking new trees, and at the same time you had development of the new generation and potential second sister brood from the logs infested, trees infested in June, and also in August, so it can be quite messy with the phenology. And this work is published some time ago. So the conclusion of this is the part of the first parental generation engaged in late summer flight activity in Scandinavia. We have sister broods. And some beetles flying in early July shows that we have at least the initiation of a second generation development. So then we had this severe drought in 2018, which reduced the tree defenses. And this has been the most historically severe outbreak in Sweden so far, with more than 25 million of cubic meter being killed. After the storm event in 2005, we had about 3 or 5 million cubic meter being attacked, and before that it's been on a very low level, maybe 100. So what I have been asking is is the tree defense of Norway spruce recovered after the drought in 2018? And the practical aspect is, can the choice of plant material for regeneration affect Norway spruce defense against the spruce bark beetle? So my hypothesis I set up was, is there any site-specific differences? Do trees at the site with most severe drought in 2018 have lower defense capacity? And also, is there a difference between provenances? And I compared Swedish and East European provenance, which with generally early and late timing of bud burst, but there was a big overlap there in the Crutch values. And also we looked at the seasonal differences of this, and by doing inoculations of blue stain fungi throughout the whole season. And it was performed with, I inoculated trees and measured inoculation. And this could be a proxy for defense towards the bark beetle. And this is associated with a bark beetle then. And as I said, I did this on 2 classes of provenances. And it was conducted on 3 field sites which had different differently affected by drought. This graph shows SPEI values of drought index April to August from 1950, and the Skärsnäs, the most southern location, had we wanted to know if trees were more affected by the drought at that location compared to the others. So here is Skärsnäs, and the other locations were one close to Uppsala, where I work, and one up north in Norberg. And as you can see, there was a precipitation deficit in all places, but Skärsnäs had the most least precipitation during this year, especially compared to the average value. And also thermal sum in all locations, all the sites were way above average. So the results I got was, now the dry site did not have lowest defense in 2018. It was rather Lugnet, the one close to Uppsala, and the seasonal aspect, that apart from Lugnet, you had the highest, biggest lesions and lowest defense in mid-June, which is the time when bark beetle activity is most they're most active, and which also coincides with the sister brood flight. So, but it was rather on a shorter term we could see relation to precipitation. So the 2-month rainfall in the preceding 2 months correlated quite well with lesion size. Another thing I noticed at the most southern site was we had differences in site conditions, in soil wetness or soil moisture. So we had some trees growing in and some, and I could drier soil conditions, the lesions were much bigger. So the conclusions from this after 2019 was there was no clear drought lag effect and stronger correlation with the 2-month precipitation. And we could not either see any differences in tree defense between the provenances. But we could see this higher tree defense in trees growing on the wet soils. And then in 2021, I received more funding to redo the trials in the same procedure and to look at year-specific differences and see if there is a higher tree defense capacity in 2021 compared to 2019. And also look at the within-site differences in soil moisture conditions. So, looking at the seasonal aspects and between-year differences, we could see that all sites have like a similar profile of tree defense in the season, apart from Skärsnäs having this, which I can't explain really and haven't found out why. But you see that the most activity is still in June, and that's where we have the sister brood and most drought stress is starting. To the within-site differences in trees growing on moist soils compared to dry ones in Skärsnäs. But this was not as clear in 2021 as 2019, as I saw earlier. And comparing it in Norberg, we had the opposite way, comparing intermediate conditions with dry. Then there was lower defense, higher defense at dry conditions. And doing an ANOVA analysis on this, with also considering the diameter of the trees, there is some significance. However, looking closer to this, in Norberg there is the same pattern regardless of the tree size, but in Skärsnäs the significance is not there, and it's even the opposite way in 2021. So my final conclusions is that high tree growth, low tree defense, high beetle activity in early summer means high risk for successful beetle attack, and there's a complex nonlinear relationship between tree defense and drought stress, and partly in line with the growth differentiation. Thank you. And this cute little fellow says, "I'm never going to murder another spruce again." and also thank today's people involved in the project.
Thank you, Peter,
Thank you.
to fit to the time.
Yeah.
So, other speakers coming, and thereafter we will have the opportunity to make questions to you. Thank you so much. So, next speaker will be Rafael Zas, and we'll talk about the G-by-E interaction in defensive chemistry in maritime pine.
Okay, hello again. I am Rafa Zas from the local institution, Emisión Biológica de Galicia, and I'm going to keep talking about the effects of the environment on the genetic evaluations related to resistant and defensive traits, a topic that we are seeing in the last talks that could be very important modulating the outcome of any breeding for resistant activities. Well, during these days we have been attending many talks that have led clear, well, this is too much, that our forests are exposed to many sanitary problems all around the world, and that I think all of us are good defenders of, strong defenders of breeding for resistance as a key tool to try to deal with this important challenge. Most forestry species has enough genetic variation within intraspecific genetic variation that can be exploited quite easily through selection and breeding to deploy resistant material that can be performs much better under this context of rising biotic risks. But all of us, I think, that are aware that breeding for resistance is not easy, and it's full of many challenges that we need to solve to be successful. And there are many challenges that we can spend a lot of time talking about them, but I am going to focus on these three that will act as an introduction of my study that I am presenting. The first one is related with the talk that Jordi gave to us. It's related to the integration of all the vital functions that a tree must do to complete its life cycle. And everything is related, both from a phenotypic point of view. All functions need resources, and resources are limited, so if you invest in defense, you may not have resources enough to other functions. Multiplayer not only have led to differentiation in these different functions, but also to complex covariation, as Jordi told us, among these functions. I am focusing in the point that was lacking in the Jordi talks. Here is the same study that Jordi explained, but I am including here the differences, only the constitutive ones, not the induced ones, but we can see that there is an important differentiation in all these functions, including references, but more importantly, there is a clear geographical pattern of the distribution of these functions along the distribution area of the species, with provenance origins of prioritizing one of the functions. They do it at the expense of doing less in the other functions. Okay. The second point that I would like to highlight is that we have been talking a lot these days is about the dynamics of the biotic bricks that are affecting our forest. Most breeding in activities today are focusing on the pathologies or pests that are currently affecting our forest, but we know that in the future, in the near future, we will have probably many other problems, both because of the climate change and the shifts in the distribution of the pests, but also because of global trade and the movements of pathogens across biogeographical regions. This is a quite old figure, but it illustrates pretty well this. This is the evolution of the number of alien pathogens in Europe during the last years, and it's clear that the evolution follows an exponential trend, and that we are supposed to be exposed to new problems in the near future that we don't know who are they. So with this in mind, I think that focusing on things like investment in resistant traits, in general resistant traits that can protect the trees against pests and pathogens in general becomes quite important, and that is what I am doing here in the talk. And the third point is related with the impact of climate change in the host defenses that we have seen in these previous talks. I think most of the resistant traits are plastic to environmental variation, so they are supposed to be plastic to climate change, but we still know little about what is the interaction between how this response to climate change in relation to defenses may differ or not among genotypes within a species. And all these 3 points becomes even more important because most of the efforts that are being carried out for breeding for resistance are usually focused on a single pathogen that is currently affecting our forest, and the screening protocols are usually or typically done with very low or environmental variation, or even under artificial conditions using young material that may drastically differ from the real conditions of the forest. So the objectives of the study that I am presenting here is to quantify the genetic, the environmental, and the G/E interaction effects on defensive traits in maritime pine, and to explore patterns of covariation of these defensive traits with other life history traits such as growth or drought tolerance. I am doing this in maritime pine, which is a pine forest in southwest Europe and also here in Galicia, which occupies a large area in this area with many small and isolated populations that are very differentiated in many adaptive traits, and the range of the environmental variation within this the distribution area is quite large, from the very good conditions, good climates where we are now here, to very dry environments in the south, in Morocco, and it is in other traits than the fence. So we take advantage of a net of provenance trials that includes 10 contrasting populations of the distribution range of the species, the figure in the right, and they were established on common gardens, in 7 common gardens in the area of Galicia. all the populations were included in all the trials. So when trees were 13 years old, we sampled needles, branches, and stem phloems, and measure a battery of chemical and physical defensive traits, including terpenes, phenolics, tannins, and also some physiological traits related to growth, to the physiological status, and to the nutritional and water status of the trees. I am going to be quite quickly through the results. In this big table, I show just the results of the univariate analysis of all these single traits. And the important point here is that besides genetic differentiation in most of the traits, which is the column in the left, the genetic effects, the important point is that the environmental effect, the plastic effect to this variation across the 7 environmental tests was highly significant for all the assessed traits. And also, there was significant G/E interactions in many cases. Let's go a bit further. And in this figure, I try to quantitatively compare the environmental and the genetic effects for all these traits, defensive and related to growth and physiology and water use efficiency. And the important point here is that the environmental effects dominated over the genetic effects for almost all traits, and especially for those related with growth and physiology and the branch and stem defenses. When we do this comparison, looking more in detail to the profile of terpenes, the genetic effects became greater, and with some particular terpenes being showing a strong genetic differentiation, which is consistent with the known difference among populations in terpene concentrations, which are good fingerprints of genetic variation in this species. But the important thing is that the environmental effect was again important in almost all cases. And regarding the G/E interaction, we found significant interactions for many of the defensive traits. Here I represent one of these cases to try to emphasize the relevance of the interaction, which means this is for the non-volatile resin in the stems. These are the 7 environments, and each line is one population. These are a typical reaction norm plot. And, well, as you can see, there are important rank changes across environments. I highlighted here one of the populations in red. You can see that it's the best defended in some of the environments, but is among the least defended in the other environments. So it depends a lot where you are testing this material, this genetic material. The defensive performance will be extremely different. But when we have a look to the complete G/E matrix of all the defensive and physiological traits, and do just a simple principal component analysis on this, you can see integrated trees. Each color in this figure is one site, one of the 7 testing environments, and you can see clearly that they appear aggruppated by sites, defining some kind of syndromes, as Jordi told before, in relation to the investment in the different life functions and defensive strategies. And this is even more important if we take a look to the environmental variation that we have in our network of common gardens. This is the climate envelope of the whole distribution of maritime pine. These are all the gray points. And these are our 7 environmental plots. So we are only focusing on a very small part of the environmental variation where the species can grow. And in the speed of that, we found that the environmental effects were extremely high, modulating the investment in defense. And these are, in fact, the conclusion of this talk, that in summary, despite low environmental variation across the test environments, environmental effects dominated over genetic differentiation for almost all traits. The G by E was also relevant for many traits, and the environment appears to define some kind of defensive syndromes that integrate with other life functions. And that's all. Thank you.
Thank you, Rafa. So let's welcome to Jin Shu.
Okay, I stand here. Okay, I'm too short for that stage. I want to disappear. Oh, I cannot see the slide. Okay. It's okay.
Back, forward. This is the light.
Yeah.
And let's check the slides.
Yeah, I just read slides. But I can start to introduce myself. So my name is Jing, and I'm from University of Copenhagen. So today I'm going to talk about one study we are working on now to understand our enemy, trying to find a solution how we can manage it better. So yeah, better way to slide. So it's more interesting than just standing here talking. Here? Okay, perfect. So the enemy I'm talking about. It's not moving. Yeah, now it works. So the enemy, as I said, the Adelids nordmanniana, also called silver-fur woolly Adelid, naturally co-occurs with Nordman fur in the Caucasus region and was introduced to Central Europe in the late 800s, together with the imported Nordman furs. So Nordman fur is the most popular used Christmas tree species in Europe. Denmark, although it's a very small country, but it's actually one of the biggest producer of Christmas trees in Europe, with annual production of 12 million trees, which equals to approximately 240 million euros. So it is a very important economic trees. However, so when you introduce a tree species and you together introduce your enemy, and since the lack of the natural enemy to the Adelid, so it's turned out to be a severe pest in the Nordman fir plantations. And it mainly attacks the young shoot and the needles. So typical symptoms is the curled and discolored needles. So Christmas tree is a special forest product. The tree itself is the final product. How it looks decide if it's valuable or not. So with damage caused like the symptom in the picture, it's not sellable anymore. So about 40% of area in Denmark, like Christmas tree plantations, has to use chemical like pesticides to reduce the economic loss, which is not environment friendly. We have a project for 4 years now trying to develop organic Christmas trees. So we, of course, as a geneticist, we have belonged. However, we don't know so much about our enemy, very little knowledge. So that's also one our objective now, to try to develop our understanding of these aphids, especially the population development and interaction with the different weather conditions. So at the end, of course, if we understand more, we can promote like a non-chemical treatment. So one of the environment factors we study is to understand what the different light intensity impact the population development. So we use 196 trees from 20 somatic embryogenesis clones. Those are the potted trees. This like a commercial plantation, they only sell the potted Christmas trees, which allow us to have more controlled study over here. So we randomly distribute these 196 trees in 3 treatments and repeat into 3 blocks. So the 3 treatments, as you can see here, basically represent different light intensities. So the open air are controlled and within decreasing of light intensity in another 2 treatments. So after established the treatment, we infested all the trees with adelids we collected in the natural infested clonal seed orchard. And in the middle of the tree, you can see here this temperature sensor. We also keep tracking on the temperature under each treatment. So after infestation, we keep tracking on how the population develop. And one of the methods we use is to use a score system from increasing of the density to count the aphids. It's driving me crazy, but I haven't worked there to open that data yet. It's really amazing. We actually managed to do that several times. So later on, we will have some results. But today, I will only present these results. It's basically the same thing, but not detailed, real number, just a scale. So in addition, we also look at the trees. We want to know the flushing time of the trees. We use this flushing score has been for a long time in our breeding program. So also want to study if the different light intensity impact the flushing time of trees. So one of the results, of course, from the genetic point of view, and same like our previous study, there is strong genetic variation for the resistance to adelids. So the broad size heritability is 0.25, which is quite promising for resistance breeding. We expect a gain out from the selection. So those clones here are ranked by the predicted clonal values from the largest to smallest. So those clones at the bottom are the most resistant clones. And look at the red numbers. Those are the predicted flushing scores. They are almost 0. They measured at the end of May, which is quite late for Nordman fir. So it means those resistance clones also flush very late. So that's later on I will talk a bit more about this part. So yeah, so here, so is this resisting or skipping? So this year and 4th of May, I observed that the aphids already at the stage of laying eggs. So in spring, the adelids also have a different life stage. So the final stage, like before the crawlers moving on to the new needles, they start laying eggs. So they already all laying eggs in the beginning of May. And for the early flush trees, they are just ready. The new needles, working now? Still, yeah. The new needles are ready to feed the new aphids. However, when those trees flush very late in the May, they are still haven't brought breast yet. So the crawlers, I don't know if you can see these black dots. They are from the eggs. They're the turtle crawlers. They should move to the new needles to feed to have nutrition there. However, for those late flushing, they're just sitting on the old needles and waiting for food. For such a long window, if they cannot hold on anymore, then they just die. So that's why I'm talking about. Is it resistant clones? Are they really resistant, have genes, or they are just simply because they skip from the aphids? Oh, too fast. So then also look at different treatment. Does the light intensity affect the amount of aphids? And the answer is yes. We find a significant difference over the amount of aphids under different light treatment. So in the high shade, which this bar has the highest amount of aphids on average compared with open air. So on the left, that's a difference of the light intensity we measured under the treatment. So can we just conclude that adelids or aphids prefer the shady spectacle or careful about our conclusion? Because look at the design. We are not only creating shade. We put some layer of net on top of the trees. Of course, we reduce light intensity, but we may also risk of introducing some other nested treatment factors. For example, maybe temperature, of course, reduce the rain, and also we actually delaying the development of adelids. So as I told before, I also keep track on the live development of adelids in the spring. So after over winter to become swollen, become a ball, and then laying eggs. So this is the S4, so the final stage before crawlers. So we measured at the same date, middle of April. For the open air, the yellow bar, that's so more on average more than 50% of the adelids in the trees in the open air treatment already produced eggs. So however, for the other shade treatment, they are mainly in the stage 3, so they're about to produce eggs. So it seems like the shade treatment actually delays the development of adelids also. Then we also look at if the shade treatment delays the flushing time of trees. Probably because it's too short time, it's only 1 year study so far. And we didn't see significant impact of light intensity on the flushing time. How about temperature? Luckily, we didn't find significant daily mean temperature among different treatment. However, they seems like have a difference over the temperature range, which is the difference between the maximum and the minimum temperature per day. So the green line are the shade high treatment. Seems like it has a less variation of temperature. So it seems like, maybe adelids also prefer more stable temperature. Yeah, which I could not separate from the other results, but that's just one of we can explain. Then, of course, we also find a significant difference over the Monterey from open air with the other high like a shade treatment. So the shade high has almost no rain going in, although it's just 2 layers of net. So it's impressive. So that's all from my study for now. And I will not repeat the conclusion I just said. So it's really just a study we are working on now in the middle of the project. So we have still 2 years to go, and there's many more things we need to explore if we want to understand our enemy. So if anyone has any experience or suggestions, I would love to hear that. Okay. So the last page is the people working this project and also the funding and the host. Thank you.
Jean, thank you so much, and thank you for adjusting to the time. Actually, thank you to all the speakers. And questions, but I mean, because I'm the moderator, there's a question for Jean that I cannot wait to the end. I mean, have you think about bird predation because under the shade the birds cannot go to eat the insects. Can be this helping to defend trees? Yeah.
Works? Yes, that's a very good question. I think that's actually one of the notes we try to make when we evaluate the trees. We look at the predators, for example, other insect. Of course, birds, because you look at the tent, seems like for birds such a big is maybe less easier to move in, so it have a less chance to be fed by the birds. But for the other insect, it doesn't seem like has difference. So the ladybugs also are over in different treatment. Yeah, but that's
Thank you.
definitely.
Yeah, it's working. So let's start with the questions. Sí.
I have a question. Janita Hajabdich, Giri, University of Tennessee, for Rafael. So the struggle is real working with trees. So inoculation is hard. And do you think you would find really big differences if you completed the same experiment in the greenhouse? And how would you see those shifts in both genotype and environment? If that makes sense.
Well, in my experience,
Please.
I think that I repeat some experiment with genotypes, provenance, families, or whatever in different environments, the outcome is a chaos. Yes. So and if you compare little seedlings in the greenhouse with mature trees in the field, it becomes even worse. And then the solution, I think, is to in our evaluations of resistance. Just try to cover more variation in the environments. And to check whether probably the species that I am working with, maritime pine is particularly plastic, and there is a lot of variation in plasticity in this species, and maybe it's not the same in others. But we need to check these issues before deploying material based on just a single screening in the greenhouse, I think.
Hey, it's Marcin from University of Tennessee. I have a question to Somia. In your experiment, did you use plants that are clones? In other words, have you accounted for the genotypic variability?
Yeah, so thank you for that question. We actually used a mixed population, so of trees irrespective of their genotype or without any background information of knowledge about resistance. So we procured the seedlings from a nursery. So we used basically the same set of trees that are planted across US and where we see disease developing regardless of genotype. And to my knowledge, the pathogen, it's very fair. It doesn't go by genotype, but yeah, that's...
Okay, and the follow-up to that is whether in your experimental setup have you accounted for or have you analyzed any volatile terpenes that could be affecting or changing your pathogen essentially as a defense mechanism, because you seem to be observing that with the RNA-seq?
Yeah, again, a great question. Yes, we have looked at the involvement of terpenes and volatiles in defense against the same pathogen, not in the same study, but in a different study, which I would be giving a presentation this afternoon at 3. So yeah, I'll talk more about that.
Thank you. Yes, this is also for Sumya. Did you look, when you did your temperature experiments, did you look at a constant temperature or day-night change,
Yeah, so the temperature was a day-night. Our main focus was to have the range, a higher range of daily temperatures, starting from night temperatures of 18 degrees to 33 degrees, instead of the average normal temperatures of a day, which is from 15 to 28. So we looked at the entire day temperature.
And have you considered adding carbon dioxide, increased carbon dioxide to the mix, to experiment?
Well, yes, but with the conditions that we already applied, the trees were pretty much dead. Like, I already presented results for trees that were affected with Diplodia scribiculata, and this experiment was done in 2016. So, do you want to add more, Enrico?
Yeah, I want to add to that because he came in at a later stage. So we actually discovered that because the environments are so contained, the pines were sucking up all the CO2 during the day. So we had to actually supplement the chambers with extra CO2 to get up to about 350, 400 ppms. Was that your question? Yeah.
And even more so due to climate change, from what I've read through the daily journals.
Ben Bumner, Twin Institute of Forest Genetics. The question is to Ying. How have the Nordmann firs been cloned? Is it cuttings or is it?
Somatic embryogenosis. I use somatic embryogenosis.
Okay, so these are special genotypes. Is it true? Because normally not all genotypes can be cloned. So you know something about the genotypes? They are special?
Well, I mean, in Denmark, actually, one of our past colleague, Jespen, he started this somatic embryogenesis clones. That time, he's not a breeder or geneticist, so he just want to test the method. What he did, he just used the seed directly imported from Caucasus region. So those are random selected, like, even not selected, just random trees from direct import. So they are not special clones.
Yeah, thanks.
So I have a question for Rafa. And from your presentation, it seems like environment really is the major driver here as far as defenses are concerned. Should we even care about understanding tree microbe interactions on the genetic level? if when you put them out there, it doesn't matter, really?
Here, no, because probably you throw me out from this group. But besides an environmental effect, there was also a genetic effect that is still there. Is that what you mean?
But it seems like the environment really kind of washes over the genetic effects in some way.
Well, it was greater, clearly greater than the genetic effect in this particular case, at least. But I think it doesn't wash everything. I mean, some genetic effect is there, and that is why breeding works. But I think this is also happened, for example, with breeding for higher growth. You can see interactions, G by G interactions everywhere, and even with these big interactions, you are able to improve growth a lot.
Just a quick follow-up. Even in agriculture, people work on like this single resistance gene that works excellently well in the greenhouse, and they plant the stuff out, and it breaks, and that doesn't work. You know, different conditions of temperature, humidity, what have you, the resistant genes goes out the window. So, yeah, it's just something to think about. Yeah.
Actually, I would like to say something. We don't know how much genetic variation is a lot. I mean, same for evolution. I mean, environment is always there, but how much genetic variation is a lot or is a lot? Looks like it's enough to things happening, right?
Yeah, okay. I wanted to maybe just comment on Enrico's question and the response. As Raf said, height is under very high environmental control, 90% versus 10% genetic, but it's the #1 trait for breeding, and it's been very successful. So high environmental variation doesn't mean you can't get genetic gain, but it's great that we're looking at this and trying to understand it. It's essential. If you look at the literature when it comes to defense traits, especially terpenes and phenolics, they're much more heritable than anything else that breeders usually look at. So I think you have to look at measures of heritability. That's what will count in the end. Thanks.
So I would like to thank you for really nice presentations, and I really liked your presentation, Rafa, but I would like Petter to comment on his sites in relation to Rafa's presentation, because you were looking at one provenance trial, were you not? So then I assume that 2 of the sites were within the intended range and 1 was under stressful conditions. Is that correct?
Once again.
Okay, so you were the provenance trial you were looking at. As I understand the Swedish breeding program and the testing, they put 2 of the sites within the intended range and 1 within the future range. So which one is in the future range? Is that the one that was the outlier site, Skärsnäs?
We used, I don't know really, but we used same provenances at all locations, and they were of either Swedish or East Latvian origin, and they were the same ones in within year. We used the same provenances, but between years there were not enough trees not inoculated.
Okay.
So that was the setup.
Thank you.
Does it work? Yeah. It's a question for Rafa. The results you have presented are for provenances and for different, it's a multi-site study for provenances, and the results show that environment is especially important. Do you think it's going to be the same if you work within a provenance, such we work in provenance in breeding? We usually work for a provenance, yeah, and we have several families or several genotypes within a provenance. Do you think it's going to have the same pattern, to follow the same pattern?
Well, if you think in these results that are based on provenance, the G-value interaction means that the response to environmental variation varies across populations. So the answer to your question, it will depend on which population are you studying. If you are studying a population that is especially susceptible to environmental variation, it could be also variation. I think there will be more chance that there is also intraspecific population variation in plasticity. If you are in a population with low plasticity, the chance is lower, I will say. But probably it's not that simple. because it will depend on the variability of plasticity within the populations, which is probably different among populations.
So we should study that part.
Yes, I would say yes.
Okay.
At least trying to find the good result and is not important, so you get very happy.
So can I make a quick follow-up question on Raquel's question? So how many different environments do you think we should be testing the trees in order to get a good estimate?
More than 3?
Thank you. I'm Barley Rose from Oxford. Rafa, you've looked at environments. Did you look at environmental variables to see between these ones?
Not really yet, but these 7 sites are extremely close to each other, and they are supposed to be, you look to the environmental, to the climate, for example, in these 3 areas, they are the same, at least in comparison with the whole climate envelope of the species. So they may differ in the soil, they may differ in the silviculture or something different, in the rock, but not yet. I don't know which are the drivers, the environmental drivers of this variation.
Thank you.
So I think this session is over. Let's thank the speakers for their amazing talks. Thank you for the very good questions, and I believe the organizer wants to allow us to go eating. Maybe you want to say something?
No, it's just time for lunch upstairs. After lunch, we will have a short poster session, and we start with the following session. I think we have 1 hour 45 minutes for the lunch, so we can delay maybe 15 minutes the lunch, or what do you think? Is 2.5? Hi guys, we are going to start with the poster session with our colleagues from South Korea that couldn't present yesterday. Okay, so you can please come here for presenting your posters. And then we will have some few minutes for having a coffee and sit down for the afternoon sessions. Yes, one by one. No? You have, yeah. Thank you.
Okay, good afternoon, and now I'm going to start my short presentation of my poster comparing the outbreak of larvae of Pyralidae in seed orchard and natural population of Abies nephrolepis. So Abies nephrolepis is a conifer vulnerable to climate change in South Korea. As climate change is causing novel tree-parasite interaction, it is important to understand those novel relationships for the adaptation to climate change. So my study aimed to understand the emergence of insects in seed orchard and natural population in South Korea. So the study site could be divided into two. First, seed orchard, which is A in the map, is located at about 55 meters above sea level. In this site, we collected 80 cones from 38 mother trees. On the other hand, natural population, which is B in the map, was located at about 1,500 meters above sea level. In this site, we collected 93 cones from 31 mother trees. With the collected cones, we did the cone analysis following the guidebook of USDA Forest Service, and insect was identified using morphological characteristics. As a result, we found that cones produced in seed orchard was bigger. However, they produced much less pure seeds with higher damage rates. While there was no apparent insect damage in natural population, 95% of the cones in the seed orchard was damaged by insects. Various insects was discovered in the seed orchard. However, one of the most interesting facts was that we found that an isolated cornworm was eaten by pre-invaded parasitoid. So the cornworm, which we identified as larvae of Pyralidae, was the most prevalent and damaging insect found in 61% of the cones from the seed orchard. We suspect that the higher temperature and higher density of trees might provide a better condition for insects to invade. And so if the temperature was the major cause of these differences, considering the current climate change, we are afraid that these insects found in the seed orchard could possibly attack natural population in the near future. So as a conclusion, we think that the insect control in the seed orchard is necessary, and also the regular monitoring in natural population seems essential to prevent the outbreak of these insects. Furthermore, for future study, we need more detailed study considering the parasitoid and cornworm interaction, as parasitoid can be used for biological control with high possibility. And we also need to use the DNA work for the better identification for the insects. So this is the end of my short presentation. Thank you for listening.
Very nice time.
Hello, everyone. My name is Hejin Lee from Seoul National University. Today, I'd like to talk about soya aphid damage that I observed while monitoring soya corensis. Soya corensis is subalpine conifer of Korea. IUCN evaluated it as a vulnerable species because of its small distribution area and distinct decrease. Soya reproduces not only sexually but also asexually, forming lamettes. Recently, on large scale, aphid infestation was observed in the site. Though it was not identified, it seems to be one of Cynara genus species because of their large size of the body and color and the shape. So research questions are, first, is there a relationship between tree decline and aphid infestation? Is there clonal specificity in aphid infestation? Next, this research was conducted at 1,530 meters in Mountain Hanbeksan in Korea. This site is the second largest site population in South Korea. I sampled a tree every 5 by 5 square meters and investigated total 174 trees. And then I evaluated tree decline and aphid infestation from 0 to 3 levels following these guidelines. The level 3 was the level 3 is the worst. The result is as follows. Aphids were found in 69 of 174 trees. The most aphids were observed in the marginal trees marked in red. This shows that aphid migrating from the edge of the population to the center. On the other hand, severe decline trees were at the center, marked in the red also. So maybe by the candle rank correlation test, there was no correlation between aphid infestation and tree decline levels. Next slide. 49 clones were identified, but there's no clonal specificity in aphid infestation. As you can see, the aphid infestation was distributed evenly among clones. This is the end of my presentation. Further study on aphid life history is needed. Thank you for listening.
Good afternoon. The Republic of Korea. The title is Resistance and Sensitivity to Inoculation with Pinewood Nematode of 6 Pinus species. Pineweed disease is caused by pinewood nematode, and it first occurred in 1988 and continues to cause great damage to pine forests in South Korea. And my objective is to compare the sensitivity and resistance of the 6 pine species by inoculation of pinewood nematode. Experimental materials are 4 native species, and to introduce species. More than 20 seedlings of each species were transplanted into the experiment forest of Seoul National University. And high pathogenic pine nematodes was mass cultured in BC bacterium medium. 10,000 pine nematodes were inoculated to each species, and water was injected into the control group. For observing the phenotypic change, photographs were taken at 2 weeks intervals. All seedlings were collected after 23 weeks, and the total number of pine nematodes was measured by extracting them for 24 hours using Fenner methods. As a result of phenotypic observation, the infection rate was high in the order of Pinus stambergi, Pinus coreanensis, and Pinus densiflora. And table 1 shows the summary of the result. And as a result of measuring the pine nematode concentration of infected seedlings based on the dry weight and fresh weight, the average pine nematode concentration was high in berries, as the figure 6 shows. And these are conclusions. The introduced species were relatively resistant to pine nematodes compared to native species, and the death rate of infected seedlings varied among the species. And this result would provide basic information in selecting species and individuals resistant to pine wilt disease in pine species. And I think, furthermore, it will be necessary to find out the genetic effect of resistant traits in further studies. Thank you for your attention.
Okay, so over there, I was thinking on make a stop for drinking the coffee, so you can go and take the coffee, but we are going to keep going with the going to.
So this next session, it will be mostly focused on mechanisms of resistance. We have 4 speakers, 15 minutes per talk. We'll have questions at the end. And the first speaker is Nadir Erbiljan from the University of Alberta. The title of his talk is, Does Carbon Allocation and Mobilization to Chemical Defenses Affect Tree Survival During Biotic and Abiotic Stress?
Thank you very much. I'm not sure I'm going to beat up the previous 3 speakers. They did really fantastic job within 3 minutes summarize their research. So I will do my best. Here's a joke for you before we started. You know, every time I went to that male's restroom, in the sink, there are some green particles over there, right? I thought, who washed the lettuce or something? It turned out to be the paper. What? What happened? No? It's not working? No, it's okay. I'm going to stay here. I'm going to stay here. As long as it works, no problem. Okay. This is a collaborative work between my lab and then the Tom Kolb's lab at the University of Northern Arizona. This paper that I'm going to present has a story, really. The story is that I was one of the reviewers of the paper. They submitted in the Chemical Ecology, Journal of Chemical Ecology, and then after paper was accepted, I approached them, do you have more tissue samples? They say, yes. This is after it happened. So I haven't involved in the study design. You will see it's a really fascinating study design. It's very labor intensive, but they did a good job. This is about the forest decline, right? I'm not sure in this part of the world, but in North America at least, we know forest decline is a really big concept. Goes all the way from 1990s, that Manning's forest decline concept really links with, identify the complex interactions between biotic and abiotic factors. In terms of biotic factors that they promoted, they suggested that actually drought may be the driving factor for insect outbreaks. So a lot of research really focused on that drought aspect, how the drought affected tree physiology, right? Here is a try to summarize from entomological perspective. First of all, this prolonged drought might affect the stomatal conductance of the trees, right? That's pretty much everybody reported here and there. And then there's another aspect of drought that they may affect the hydraulic failure due to embolism on the xylems, may cause the disrupt the water flow. That is very important. Two aspects of the drought in plant physiology. And then we have, changes, reduction in the photosynthesis. What happens, they may deplete the carbohydrates, right? Reserves that we commonly call them non-structured carbohydrates, that really they are soluble sugars and starches. So what happens, reduction in carbohydrates may affect the mobilization and transportation of the non-structural carbohydrates to various carbon-dependent defense metabolites, such as terpenes or phenolics, that may serve as anti-herbivory functions. That, ladies and gentlemen, may affect the tree's ability to defend, basically, against the subsequent insect attacks. That may be, including bark beetles. This is a very simplistic overview of the bark beetle host tree interactions. We know that bark beetles complete their development. They are cryptic insects that pretty much spend their entire life cycle on their bark, feeding on the phloem. Phloem contains some toxic chemicals, mainly terpenes and other carbon-dependent compounds, such as phenolics. But terpenes are also toxic to bark beetles and their fungal associates. One of my students actually published a really nice paper last year that's summarizing that bark beetle host tree interactions of, in case of mountain pine beetle study system. Is very nice. I suggest you to read it if you have time. And what's missing in this field is really conducting field experiment that look at the integrates basically carbohydrates, three defenses, and changes in carbohydrate reduction until tree death. This talk is about this aspect of research. No? Okay. This is my hypothesis. I'm just underlying the drought stress, that the wording over there. My hypothesis is that drought stress trees respond to bark beetle attacks by increasing the non-structured carbohydrate allocation to chemical defenses, a response that can enhance carbon-based defenses such as terpenes against bark beetle attacks, but also deplete carbohydrates reserves and thereby contribute to carbon starvation and tree death. This study is conducted in the field, again, in the Pontevedra pine forest of Northern Arizona University. Have 2 levels of water stress, 3 levels of bark beetle exposure, and they have 8 blocks and 1 tree replicated in each block. So basically, total number of trees used here is 48, and is 8 for each treatment combination. This is the drought treatment was applied in June 2013. Control is ambient, do nothing really, just leave it as it is. The drought was induced by cutting the surface roots of the trees by backhoe, is really trenching around each tree, and they put some tarp around it so moistures and roots cannot penetrate through that system. It's very simple and very effective. And in terms of application, biological application started in July 2014. The control is, as you can see here, the trees are protected by spraying carbide on the surface of the bark, so this stopped the bark beetle attacks. The second category is the bark beetle attack was induced with the pheromones. In this case, they used the Dendroctonus brevicomus or western pine beetle lure, and they counted really every day, every other day, they count the number of attacks and they put these different pins. And based on that correlations, they also capture the beetles, right? They crush the beetles on each hole corresponding to the tree that attack in each field. So basically, this is the fungal inoculations associated with the beetles. There is no beetle involved here, just the beetle took, they crash the beetle on the floor. It's a very simple method. And every 2 weeks, what do they make? at the leaf pre-down and midday leaf xylem water potential. Also, they look at the midday stomatal conductance of photosynthesis. They estimated tree mortality, that trees are considered dead if 90% and more of the canopy turned to red or yellow, the brown. And they also collected the phloem, and those phloem tissues used for monotropion analysis. This is why I get in the project. This is what they did. They collected cores in the opposite side of each tree, and every month from July to November 2014 and May to September 2015. And we use analysis as well as a starch analysis, starch and sugar analysis. Again, this more details of this work and methodology was published in this paper. What we have, first of all, you can see the treatment combinations here: trench attack, untrench attack, untrench control, untrench inoculated, trench inoculated. In all cases, actually 2 treatments yield mortality. First one is the trench attack categories, that 6 out of 8 trees were killed within literally 2-3 months after they put the baits. And 2 of the trees in untrench attack categories were killed in the following year. There was no other mortality occurred in the study. You can see the changes, sudden changes of the canopy coloration from really in the same year, from July to all the way August, within a month, perfectly green to pretty much red crown. Okay, this figure shows the leaf water potential. You can see that the trenching was very successful in inducing stress, especially in the bark beetle attack trees that in trenched category. This is very important. Let's go back to my main hypothesis, that why I really undertook this study. I really want, this study provided 4 types of evidence that defensive metabolites become a major sink for carbohydrates in carbon-limited trees. That means that they are subject to drought. When additional biotic stresses, in this case the bark beetle attacks or fungal inoculations, demand for carbohydrate consumption, thereby promoting carbon starvation and tree death. This study, first evidence is that we found the bark beetle attacks and inoculations reduced non-structured carbohydrate concentrations relative to the control. Also, 3 treatments, we pulled the data basically in each category: trench control, attack control, inoculated control. There was no difference, and these are the starch, total sugars, and total non-structured carbohydrates. Just focus on this column. Trench has no impact, right? But attacked and inoculations reduce carbohydrate concentrations. I guess that is expected. The second evidence, exactly the same thing, but over time from July to November 2014, May to September 2015, the same categories over time. When you look at the trench, has a really minimal impact, exception of, I think, the October data. On other cases, attack and inoculated trees have less non-structured carbohydrates. Question is where those carbohydrates are going. I'm going to explain to you. Second thing is we found consistent negative relationship between carbohydrates and terpenes in both live and dying trees. Here, the top figure shows the diterpenes, the bottom figures are monoterpenes, starch, sugars, and non-structured. This is total data. You can look at the regression is about 62%. The only 6 that trees, this is based on the 2014 data. You can see the negative correlations is more stronger in the diterpenes than the monoterpenes, because both non-structured carbohydrates and diterpenes data were obtained from the cores, whereas the monoterpene data come from the phloem. Nevertheless, still significant. This is the overall pattern. Again, among all those 6 treatments, the system is the same, especially those trees that attacked or inoculated show a very strong negative response. Third evidence that we consider that terpene concentrations of dying trees were similar to or higher than live trees. In fact, at the time of tree death, that it's happened October, November, dying trees had higher concentrations of monoterpenes and diterpenes than live trees. Here, this study, this is from 2014 data, again, July to October, This the top diagram shows the total diterpenes, and this is the monoterpenes, and these are the concentrations. Red line shows the dying trees, and blue line the live trees. And you can see dying trees in both categories at higher monoterpenes and diterpenes. How about the non-structured carbohydrate concentrations? Total opposite. At the time of tree death again, the live trees had a lot of non-structured carbohydrates. In fact, when the trees die, the dying trees had about 5.8% of the total carbohydrates that present in the live trees. There's a huge carbon consumption on the live trees. I'm going to summarize everything put together. Here, try to prove that basically bark beetle attacks that on water stress trees, I would like to emphasize here, on water stress trees, rapidly deplete the non-structured carbohydrates in 3 stems by creating very strong sinks. For carbohydrates, potentially resulting in carbon depletion in trees. Some aspects of this presentation published here earlier last year, but other aspects still I'm preparing some papers. But overall, we show that dying trees continuously allocate non-structured carbohydrates to produce defense metabolites. We found a negative relationship between carbohydrates and defense chemicals. And since dying trees had profoundly lower carbohydrates but also higher concentrations of terpenes, suggesting that most likely those carbohydrates were mobilized for carbon-dependent metabolite productions. Overall, I think another aspect of drought is another fungal inclusions of the xylems that these dying trees failed to replenish carbohydrate reserves. That is very critical. This aspect is also critical. I didn't really talk so much, but fungal organisms especially associated with the bark beetles, when they infect the trees, what do? They need the carbon. Where do they get the carbon? They get the carbon either 2 sources, really, not much. Either they have to break down the secondary metabolites and acquire the carbon, or they have to attack the sugar and starch. In my opinion, at the beginning of the attacks, probably they're utilizing the sugar and starch, but later on they are able to break down the metabolites to get more carbon. That also, this process created additional sink in dying trees. Thank you very much. I am on time.
Wonderful talk, Nadir. Up next we have the Ohio State University. Is there a role of terpenoids in systemic induced resistance in Austrian pine?
All right. Good afternoon, everybody. I don't know how you all are doing. I'm still trying to settle down from the delicious food we just had and a little too many glasses of wine that I consumed. But yeah, I would try to do my best to not sway away and keep my talk strictly about if and how terpenoids contribute to this phenomena which we call systemic induced resistance in Austrian pine. So when we talk about tree immunity, it is mostly described in the form of an induction event by the pathogen, which can, which usually induces an array of host defense responses. Now, if these defense responses are able to successfully ward off the pathogenic attack, the trees become more and more resistant to future attacks by most pests and pathogen, thereby resulting in a phenomena which we call systemic induced resistance. On the flip side, if the initial induction event is so overwhelming, the host defenses may fall below the baseline level, which results in increased susceptibility of trees, where trees become weaker and weaker and more susceptible to future attacks by pests and pathogen, a phenomena which we call systemic induced susceptibility. Now, these concepts or these phenomena has been demonstrated in many different pathosystems. However, we still don't know the mechanisms that underlie the expression of SIR or SIS in trees. So in order to investigate that, we chose the Austrian pine Diplodia pinea pathosystem, most importantly because the temporal concept of SIR has been demonstrated in this system. The disease severity has been demonstrated in the form of stem lesions caused by Diplodia pinea infection in Austrian pine. The host defense mechanisms, in response to a Diplodia pinea infection has been also demonstrated through various specialized metabolites like phenolics, terpenoids, lignin, as well as we also found the involvement of pathogenesis-related proteins and phytohormone crosstalk. And then in the previous study I presented this morning, we also found out that the fate of carbon and nitrogen, and something which also Nadir talked about in the previous presentation, is really important in this pathosystem as well for deciding the outcome. So then terpenoids could be involved in the expression of SIR. And in order to prove that, we had to first demonstrate SIR or SIS by systemic lesions, and then thereby try to deduce the role of terpenoids by profiling. So first of all, we set up an experiment in 2020 where we used 4-year-old Austrian pine trees, and we inoculated them in the stem at a height 5 centimeters from the base on opposite sides using plug inoculation. Now, the inoculation was made either with Diplodia pinea or wounding, where we just used the growth media without the pathogen, or we did not induce the tree. And this acted as the induction event in this experiment. Then as a second factor, we made a second inoculation, which we called the challenge inoculation, this time only with Diplodia pinea, at a height 15 centimeters above the height of the induction event. So this was, quote unquote, our systemic location. This challenge inoculation was made, however, 12 hours, 72 hours, or 10 days post the induction event. And then we collected samples first at the time of the inoculation, which acted as the constant samples after the incubation post-challenge, which was 2 weeks, and when we also measured the lesions. So just visually, we saw that due to the pathogen induction with Diplodia pinea, the challenge lesions at the systemic location was much smaller. And when we compared this to the mock challenge lesions, it was definitely smaller. I'm sorry if it's not visually, you cannot visually tell them apart, but to further prove that, we ran an ANOVA where we do see similar trends in wounding and pathogen induction, where lesions are progressively shorter with increasing time of challenge. However, the lesions produced by pathogen induction was much smaller, kind of indicating that yes, we do see SIR not only due to a pathogen induction, but the SIR response magnifies as the challenge time increases. So having proven SIR in the system, we then wanted to look at the terpenoids, where most of the work was done in the University of Alberta, Canada, in collaboration with Dr. Nadir Erbilgin and his student Guncha. And the details of the acquisition as well as the processing is mentioned here in the slide. And I'll quickly move to what we did with the raw data of the terpenoids and VOCs that we obtained. First, we looked at the inducibility to see how much these compounds are getting accumulated based on the induction event. And we devised the inducibility based on the work that was done by Katerina in the year 2014. And we looked at inducibility along 4 different direction vectors. Vector A, where we tried to look at the the immediate systemic response due to the induction event at the challenge location. Vector B, where we looked at what's the long-term response or what happens after the incubation at the systemic location. Vector C, where we looked at what's happening locally at the induction event, pre-induction and post-challenge. And then vector D is where we looked at what is happening at the systemic location, pre and post-challenge. So just to further emphasize, we further expressed the inducibility data as percentage for better clarity and ease of understanding. So, So firstly, we see that along vector A, which is the immediate systemic effects, we do see that due to the pathogen induction, there is very high inducibility of very specific compounds, namely alpha-pinene, beta-pinene, limonene, boronyl acetate, and benzaldehyde, which is a group of monoterpenes and some VOCs, volatile organic compounds. We do see some expression in the wound-induced and the non-induced, but mind you, if you look at the levels, the scale in which these compounds are induced in the pathogen, they're 7,000 compared to the other, and I'm still talking about vector A. The levels are much smaller for the wound-induced and the non-induced trees. And then when we shift to the left-hand side of the slide, where we see vector B, or what's happened after the incubation time, we see a much stronger response in all the induction treatments, and even more so by the pathogen, where you see the same compounds expressed to a level of 80,000. So then further, when we looked at the vector C, we see the trend has sort of declined. So the inducibility of these compounds have now lowered. However, at the systemic location, we do see that these compounds continue to be induced, you know. So that was some idea going forward, and then trying to classify the compounds to further get some insights into what's happening in the system. So we used this dimensional reductional approach by Saurav Chakraborty, who was a earlier postdoc in our lab, and I'm going to talk about the individual results here on. So first we created some NMDS plots, and for the pre-challenge samples at the systemic location, we found two main groups, one which was monoterpenes, the other group which was made of the volatile organic compounds. But when we tried to look at the same samples after 2 weeks, we did not see any significant grouping. So, and then which could tell us that probably the effect produced after 2 weeks is almost same by all the compounds and doesn't have much variation with respect to each other. So to further elaborate on the findings, we created some hierarchical clusters for the pathogen inductions pre-challenge and post-challenge. And we do see, while there are some compounds such as alpha-pinene, beta-pinene, and the range of VOCs we reported earlier, that is consistently, significant pre and post challenge. We do see certain compounds such as beta-pinene, limonene that appear significant after the 2 weeks. And then when we compared this clustering pattern with the wound-induced or the mock and the non-induced clusters, we saw there was big difference between the pathogen clusters and the clusters produced by wound-induced and non-induced trees. However, the wound-induced and non-induced between themselves did not have any difference. So these clusters are exactly the same for the respective induction treatments. So going forward then, to further get some clarity, we generated some emergent self-organizing maps for the same two set of samples. And for the pre-challenge samples, we found that the pathogen-induced samples for the 72-hour time point challenge was the most significant to explain all the variants that we observed in the data set, whereas for the post-challenge, we do see the 10-day time point for a 10-day challenge. So the longest, the longer the time of challenge, the more the effect that you see in the terpenoids and VOCs. So to further establish our findings, we did a direct correlation of these various clusters that we found in HCA and NMDS, And we tried to correlate them with the mean lesion sizes produced, and we found negative correlations with cluster 1, which was alpha-pinene, cluster 2, which was beta-pinene, limonene, and the other VOCs. The terpene derivative to prove the we use the SIR and we found that alpha-pinene, which was cluster 1, can slow down the growth up to 3 days. However, after 3 days, the fungi keeps on growing on the plates. For cluster 2, it was fungi static, because when we subcultured the fungi into non-amended plate, it continued growing again. And then cluster 3 did not affect the growth of the fungi when compared to our mock, which was DMSO, and our control, which was PDA. And so we were able to make the conclusions that yes, terpenoids are indeed involved in the expression of SIR, both immediate and slow down growth, and then the other compounds are able to stop the growth of the fungi, not kill it. And then most importantly, diplodiapenia is able to use some of these monoterpenes as carbon sources. And then finally, a speculation which we hope to prove through our ongoing gene expression studies is that some of these compounds, such as alpha-pinene, beta-pinene, or their precursors, such as GGPS, FFPS, could be involved in SIR-based signaling. And with that, I would like to acknowledge my advisor, Enrico, the lab, the past members, the funding agencies, and would finally take any questions at the end of the session. Thank you.
Thanks for that great talk. We'll have questions at the end of the session. Next up is Ricardo Alia from the Consejo Superior de Investigaciones de Ciencia de España. The title of his talk is Genetic Variation for Resistance to Abiotic and Biotic Stresses in Breeding Populations of European Forest Trees.
Okay. Thank you. Can you hear me? Okay. No, here. Okay. Well, I'm going to present a work that we have been done in the framework of a European project before us. And I think that it's interesting to put it into perspective what is the objective and the main outcome of this work. First, this project covers breeding in Europe in a general perspective in forest trees, and we are working mainly in different topics of our research, mainly working with resilience, trade-offs, forest management, and tree breeding. The main part is the genetic diversity, and the talk today is about two main applications. The optimal use of forest genetic resources, that we are interested to know under which circumstances and which is the normal reaction of this improved material to be used in the European forest, and also to make better use of the genomic-based selection and other tools in forestry breeding. But of course, we are working with very different socioeconomic areas. Tree breeding, even if we talk about tree breeding in a general context, is depending a lot of the socioeconomic perspective or socioeconomic part we are working. Here we have from the Northern Europe countries with well-established and long tradition tree breeding programs, for Southern European countries, where in some cases there are 16 well-established breeding programs, and in other cases we are more focused in low-input breeding. That is, I think that we need to take advantage of the new development in genomic tools. And of course, there is a a large variety of trees. Norway spruce, Norway spruce, Scotch pine, Martin pine, poplars that they have, Douglas fir in some cases, eucalyptus that have long tradition improved programs, but even in this species is not more than two generations breeding. But of course, in other, as a stone pine and common ash, that we need to focus in other strategies. And of course, in the project, we're interested in to include in this breeding program some traits related to adaptations to different environmental and to biotic factors. And this is a quite challenging question because, of course, to include new objectives in the breeding program requires a lot of input, a lot of energies, a lot of testing, and also because we need to revise some of the theoretical approaches that we need to address. This is a question to discuss about. While in the traditional breeding, we have heard this morning about the multi-environmental trials. In some cases, independent of the resources, there are well-established breeding programs with a lot of multi-environment testing sites in the traditional breeding programs with the phenotype, in which, in most of the cases, in the height growth is the most important trait for selection in some cases, and but it was quite easy to measure, and what related to many other traits, and we incorporated other traits related to good technology or resistance. But of course, now we are in the new development based on genomic approaches, in which we are changing completely the scope. We are using breeding value based on genotypes. We are making an effort in genotyping a lot of material in order to determine which is the basis of the genetic basis and the best approach to select material for the next generation. And of course, we also need here they call the envirotyping, that is to refine the way of characterizing the environments. What is important for me in this talk, because I am not really working in biotic interactions, is that in some cases, environments, when we are evaluating resistance, is limited. We are using a reduced number of sites, a quite induced by human in nurseries or in common gardens, that is even restricted in comparison to the traditional breeding in forest trees that are really limited to plantations. And then this is a quite important question. And also because in the low input breeding, we are really focusing in including some genotyping mainly to avoid some problems of crossing and determining the genealogy of the trees, and to improve only the selection of phenotypes. Then we have now working in two types of traditional breeding, including identification of genotypes to avoid problems related to the parentage of the trees, but we need to focus on new development. This morning, Jordi was presenting this part that I think is quite interesting for us to put in perspective what we are doing. We need to change. We need to change to a new ways of phenotyping. This allows us to study many different traits probably related to adaptations, but of course to include in the breeding objective, we are in some cases losing the really meaning of these different related traits to the breeding objective. And then, in this really very simple work, we were compiling, we were trying to analyze if in the genotype selection coming days that we are now, if we are close to apply in forestries in an extensive way. There are many questions that we need to address. One is the high quality genotype is essential to success of any genetic selection, genomic selections, because we need a lot of individuals, more than the thousands, and it's not easy to obtain high quality phenotyping. We have seen a lot of discussion today, but probably we still lack some ways to have a cost-efficient way of genotyping a lot of individuals in an efficient way. Because we need to balance the cost of high quality phenotyping with the trait included in the future generation. And we need to also focus in this, if the traits we are measuring are traits that are reliable and applicable as a breeding objective. For analyzing that, we make a review, a questionnaire concerning all the partners involved in the European project to know which are the type of traits they are using in breeding populations in order to identify for which purposes are they using and in which are the main objectives and with the main advantages. We need to stress that there are different ways of approaching these traits in breeding programs. Some are based in observational studies. If there is one common garden and the pest of disease is coming or the drought is coming, they observe the effects of this event in the breeding population, and then it's an indirect way to estimate breeding value for the different families or genetic material. And only in some cases we can use direct approaches by measuring in common garden or in nurseries, inducing the response of the trees. And there is another way that we have not been talking about, but this is to use, in some cases, height as a surrogate of the effect of this event in the growth of the tree. And this is a quite complex way of measuring. Here there are some examples, for instance, in Pinus pinaster resistance, that they have been measured in different clonal tests, different traits, only to show you that, for instance, heat high, we were able to measure near 9,000 trees, but for diplosiapenia or necrosis and so on, we were only able to induce the response in near 300 trees. That means that we can estimate different genetic parameters to be used in the breeding program, but we cannot use this as a way to estimate, to apply for a genomic selection approach. And also, it's important to know that all these traits are polygenic, are really, really very high polygenic. That means that we can apply some traditional additive genetic models to improve the estimation in genomic selection programs. With as dieback, the estimation are based mainly in the effect on the crown of the trees, based on the effect of the pathogen. In Populus deltoides are mostly based in a reduced testing of different clones to estimate which are the response of the different clones to the effect. And in the case of Scotch pine, they use a reduced number of families to estimate what is the response to the otostroma, and to apply some association studies to identify some SNPs associated to the response of the trees. What is important to remind is that in most of the cases that we have information, heritability is in the order of magnitude of other traits that are important for breeding, both in a narrow sense or broad sense, and in the cases where different populations are included in the breeding population, there is a quite large genetic differentiation among the different traits. Therefore, in theory, we have really very clear traits that can be included as breeding objectives because are under high degree control. There are large differences among populations. We are also able to analyze trade-offs and so on, but of course the question is that we really do not have any practical way in most of the cases to genotype thousands of trees to apply in a more traditional genomic selection approach the traits. And of course we need a more precise definition. We do not know exactly in many cases if this correlation, juvenile mature correlation is good enough for breeding and so on. Then my conclusion is that of course we know a lot, but we know a lot for a perspective of transfer of material to identify good material for some questions, but we still lack some basic information to include most of these traits in the framework of a genomic selection approach in most of the species under consideration. Thanks for your attention.
Yes? Okay, thank you very much, and good afternoon. Can you hear me okay like this? Yeah. So I'll present results on a genome-wide association study done on ash in relation to ash dieback. But before I go into details with that, I'll give some background information. So I think most of you are already familiar with the disease symptoms. So the trees become defoliated in the crown, and trees also develop these epicomic shoots. We have these lesions on the branches, and then if we cut down the tree, we see discoloration of the wood. And there's also a lot of mortality in the ash stands. So we, in Denmark, in Copenhagen, we started out at the very beginning of ash dieback. We hardly knew we were dealing with the fungus at this time. So we had some clonal trials, sister trials, consisting of almost 40 clones, 39 clones, replicated about 25 times each at the beginning. And what we see here in this figure up here is the performance of the trees with all the ramets per genotype at one site in relation to the other site, and data collected over 3 years. And we can see that there is a very strong correlation between the two sites. Trees that perform well in Tapsø also perform well in Tusinæs. So there's, it's actually difficult to see slides. So there's strong genetic component in ash dieback disease or tolerance. Another important finding of this study was that we found this relationship between senescence or leaf discoloration or autumn coloration of the leaves. Those trees that got these autumn colors earliest had a less score in damage. So there seems to be that, like that early senescing trees seems to be less affected by ash dieback. And that made us think if it could simply be a kind of escape from these trees. So we decided that we wanted to do inoculation studies. So we inoculated branches of trees, again grown in Tusiness. And the x-axis here shows how trees perform under natural conditions, one can say, you know, when they are infected by spores. And then four trees that perform better in the trial were then inoculated, and 4 trees that do not perform very well, that perform better in crown damage, also develop shorter lesions. So that made us think, or suggest, or speculate that there must be some kind of active mechanism. It's not only caused by dormancy and dropping the leaf in the fall. And another important finding is that the trees with more disease symptoms have less reproductive success, or maybe the other way around. Those trees that have a lot of offspring, they also seem to handle better the disease. And that's especially seen for female reproductive success, which is seen here. So we see there's a correlation here, ash dieback susceptibility in relation to, or female reproductive success in relation to ash dieback susceptibility. The tendency is not as strong at the male side, but that is probably because even a very diseased tree can still manage to spread some pollen. So all in all, these different findings made us think about the future of ash. How will it recover? Are there already signs of selection in natural populations? How many genes are involved in the resistance or tolerance? Is it durable? And what is the role of phenology? So these were sort of the set-off for this new project. So 2 of the work packages are shown here. So the whole project is called Evolutionary Potential of Natural Fractional Succession Populations Challenged by Novel Pests and Pathogens. So one thing is to look at genetic architecture of tolerance, but also other fitness-related traits. And then after that, to look at the effect of the pathogen across Europe in the populations. And today I'll focus mostly on this thing here. So James Donnan, postdoc at our university, or my postdoc, we sampled 500 unrelated clonally propagated trees from different trials in Europe. And then these were also phenotyped for multiple traits, disease susceptibility and spring flushing and autumn senescence. And then he developed a genome-wide SNP dataset and plans to do these association studies. So this is just to show the scores of the bud bursts. So we have 8 different categories: dormant stage and fully developed stage, and then all categories in between. And this is the autumn coloration. So there is this clear pattern in colors, but we also have a trait that we call autumn leaf loss, and then overall average autumn senescence. But the most important trait is of course the ash dieback crown damage. So we use 2 scores. One is for small trees, and the other one is for large trees. So for small trees, if there's no effect at all, it's 0. And then if we have 10 to 50%, then class 2, class 3, class 4 is a dead tree. And for the bigger trees, we felt that we needed to have extra categories. So we have added some categories here for 2 of the other categories. And you can see we can convert actually the steps into, the scales into each other. Yes. So this is the DNA was sequenced, and then I can't really see the slide. Anyway, so they were sequenced, and he filtered away all those that couldn't map to the reference genome, and then based of those, he estimated allele frequencies and genotype likelihoods. And these, together with the traits, and he also corrected for relatedness, then this was then put into his association study. And this gave some results for the different traits. So this here shows a Manhattan plot for the crown damage score. And yeah, one would like to have many SNPs that are above this threshold here, but we don't see that many SNPs above the threshold, and that is most likely because we don't have a big enough sample size. We have 500 trees, but nevertheless we can see that a lot of things are going on different places across the genome. And then he tried to look into the 100 most healthy SNPs and used this random forest approach, a machine learning approach, as we heard about yesterday. So he wanted to, based on his SNPs, could he then predict the phenotype of the trees? And he finds a relatively good prediction, R-square of 0.55. So he did similar for all the other traits. No, I forgot this slide. So then he wanted to look into how much the different SNPs contributed to the crown damage score. And we can see that several of them are found in coding regions, but there are also quite many that are found in non-coding regions. And we can also see that many SNPs seems to be involved in the trait, and it's not that many that have a large contribution. So he did similar analysis on all the other traits, and they're actually not that different. So again, few above this threshold of 10 to minus 8, and some gives better prediction than others when he uses random forests. Here for autumn coloration we have something going on here. An autumn leaf loss and autumn senescence. So then the next thing was to try to look at how many SNPs are actually needed in order to explain most of the phenotypic variants. So he simply tried to run the programs again by increasing the number of SNPs, and we can see over here we have an x-axis from 0 to 100,000, and we can see we reach a plateau around here. So he made it with a smaller scale, and we can see that the plateau in most cases is around 10,000 SNPs. So after 10,000 SNPs, he doesn't really gain much more prediction power, even though if he increases the number of SNPs. So, but then when he then did the random forests again with this maximal phenotypic variance, the number of SNPs that are behind that, then he gets actually a better R-square score. So, so the SNPs can fairly well predict the phenotype. So the next step is then to look at the natural populations. So Katarina Budde is part of this work, and so she will get this pile of SNPs that were identified by the GWAS, and then try to see if she finds any allele frequency shifts. So we have some old DNA samples from before the epidemic, and then we have the new samples collected at the same sites. In cases where there are two red dots, that's because she went back to the same site and collected again. And those that are blue, that is new collections. And then she just wants to see if she already by now can find any signs of selection in the new generation. And she also would like to try to predict the health based on the best SNPs, or the 10,000 best SNPs. And this might be a little ambitious to look for bottleneck effects. I don't think we see that already now. So this figure here shows how she scored the trees, or how the distribution of crown damage is in the adult trees and the young trees. So the adult trees are shown in green, and the young trees, the juveniles, are in yellow or orange. And we can see that in general in Europe we find that the younger trees are less affected to the old trees, with the exception of Scotland, and that could simply be because that the disease has not been there for such a long time. So, but this is only just ongoing because she's just getting the SNPs now from James. Yes, so finally I'll just mention that a lot of people have been involved in this work. So there are some formal partners, but also a lot of collaborators, and I'd also like to acknowledge the funding agency. Yeah, thank you.
anyone want?
So thank you for really nice presentations. And Lene, this question is actually for you to start with. I might have been nodding off a few seconds or so, but what was in your 500 trees that you used for the GWAS analysis? What was the distribution of phenotypes looking in that population?
Remember this one. Can you hear me okay now?
We hear you great.
Yeah. So I haven't really looked into that, but there's no doubt that there is underrepresentation by the tolerant genotypes. Yeah.
And do you think that that has influenced the GWAS?
Of course, we would like to have a more balanced data set, and then we would probably find more SNPs that. But that's not always. That's not how the world looks like. So that was why we had to go to different trials in order to get enough of also the healthy phenotypes.
Hi, my name is Raul. This question is for Nadir.
Yeah. I was dreaming, yeah.
Nadir, if I understood properly, you show that trees dying show increased defenses. This is very, very relevant because sometimes intuitively we can try to measure defenses in the trees in order to see if they are more susceptible and correlate mortality with the defenses. In this case, this is not a good proxy, right? Why do you believe the tree is responding in that way?
I think the reason is the threat, right? The tree's priority to probably defend itself against the bark beetles. That was the concept, nothing else. Like, it's, I think, common. I'm not a tree physiologist, but I've seen a lot in the field. When last year, for instance, in Western Canada, we had a very hot temperature. So what happened? Spruce trees that they think they are going to die, so they produce a lot of cones this year. That is a reflection of because so many needles died not because of drought, because of the hot temperature literally killed the foliage, because they are green, and I haven't measured the temperature on the leaf surface area, but probably higher than ambient temperature outside. So that's a reaction. It's a reaction. They have nothing to lose, because pretty much they allocated all the carbon for the defenses, just to defend themselves.
So you mean that the combination of stresses is finally what is lethal for the trees?
Exactly, that is very important. Initial stress is important. In this case is the drought. They cannot sustain, they keep producing the carbohydrates because of limitation in their photosynthetic ability that reduced the carbon production. That reduced the carbon allocation. That affected whatever the carbon storage they have. We're doing some other work at the moment related to carbohydrates. We're looking at when the initial stress, what kind of carbohydrates they use, the one that recently synthesized, or do they use from reserves? And our data shows that they use immediately from the recently synthesized carbohydrates. They don't touch their reserves. Right, we always use the sugar first before our body touches our fats. That's exactly the same mentality, yeah.
Thank you.
So Nadir, this is still for you. Have you looked at other changes in the plants other than like the production of terpenes, like amino acids or other physiological changes in response to drought?
This is not my study. This is a Tom Kolb's study, but they look at drought-related tree physiology that published in the chemical ecology paper. But what we've done, I haven't showed the data, but this is really cool data. We have overall carbohydrate data from the roots, from the stem, from the branches, even the twigs and the foliage. I have that data, trying to put some model how the stress affect the carbon allocation throughout the systemic at the tree level. That is sort of my next paper.
Okay, cool. Thanks.
Nadir, I have a question.
Oh, okay.
So, you described about carbon allocation between structural and non-structural carbohydrates versus defense metabolites. Do you think there is possibly a role of fatty acids as well, since they're like the treasure trove of hydrocarbons and the long chains of them in fact so
They are absolutely related. There's another research that writing a paper at the moment, we're looking at how changes in soil microbial communities affect the trees' primary and secondary metabolites, and we look at the whole metabolome, including carbohydrates, fatty acids, and drive some terpenes and others. We find the significant correlations. Before anything, fatty acids always in the mid pathway before the secondary metabolite production. Yes, absolutely, they are correlated. Yeah, but I'm not a chemist either, so I'm just speculating here. Somebody might say something. I'm sure there are smart people here than me. Yeah. People are satisfied, I think.
Well, I have a question.
Oh.
And maybe this is a stupid question, but I was told there's no such thing as a stupid question. So your drought.
Are you looking at me or?
Yeah, Nadir.
Oh, okay.
So your drought treatment in this study in which you collaborated with and are presenting the results of the study that was designed by someone else. In this study, the drought stress was induced by cutting the root system, right?
Cutting this, well, I'm not sure they cut the root system, and it's about 5 meters away from the tree stem. Probably they harm the roots, but the purpose is the water won't flow from outside to the tree system.
Okay.
I bet they harm, it's about 50 centimeters in depth. Right, so there's not much water movement.
So it's
from
limiting
outside
the water that's moving
exactly.
from the outside.
That's exactly. Yeah.
There's still water that's coming down the
top.
tree.
Oh yeah.
But I was just wondering, because I could imagine that like harming the root system would also potentially both cause drought stress and probably
physically hurt.
Be a, I mean that's. It's physical damage to the root system.
It is.
Which I would think, we hear a lot about synergy between below ground stress in pines and bark beetles. So I'm just wondering if, I'm not trying to like throw a wrench in anything, but there's just something that
doesn't matter. Throw as
came to
much
my
as
mind
you can. It's
immediately.
already published. So what can you do? No. This is the point. Yes, it is probably harm, but remember, the impact may be minimal because all the measurements related to photosynthetic ability or the water flow moment in the tree was no different between control and untrenched versus trench, suggesting the trench may not have a strong impact. And we didn't find any effect on the carbohydrates either, except one area. Yes, it did harm probably, but maybe minimal. I cannot speculate on that one. It is what it is. It's not as stupid. It's a good question. I like that. Thank you. Oh, okay, I will tell Tom. All right.
No more questions?
I thought Louis is going to ask a question.
Okay, I'm Yang Gil Kim from SNU. So I have question for Gosh. Yes, you mentioned about the SIR. So your research maybe fungus? Was it fungi or?
Yeah. So we used the results. The results actually show the responses of the host to a fungal induction, a wound induction, followed by a subsequent infection with the fungus. So it's like, how prepared is the tree when it is induced with a pathogen to a secondary infection, or how prepared is the tree when it is just wounded to a secondary infection? And those
Okay, my real question is, I am studying about the nematode. So do you have anything, maybe the same happen in nematode, or maybe your opinion? I want to just...
I have no idea about nematology, so if I even try, I think people who do work with nematodes are gonna come at me. So, do you have any idea of nematode responses?
Okay, maybe I can try.
Yeah, please.
Thank you.
Thank you for the question.
Thank you so much. Yeah, hi, my name is... Can I? All right. Romulus from Royal Botanic Garden. Regarding the ash work, maybe I missed a slide perhaps, but are you looking at your GWAS, any of those were related to ash dieback in itself, or just the phenotypes? Were you trying to predict the disease, doing GWAS with case controls, or just focusing on the phenotypes?
Just focusing on the phenotypes. We
Okay.
haven't done any experimental tests, if that's what you mean with the fungus. Or-
I can hear you.
It doesn't really work. Like this? Oh my God. Okay, so we didn't do controlled inoculations, if that is what you mean.
No. What I mean is that were you looking at associations between genome and ash dieback, or you're looking at just phenotypes, GWAS and phenotypes?
But I mean the phenotype for ash dieback. Or
Yeah.
what? Not the fungus. What do you mean? I mean we look at the phenotype, ash dieback phenotype.
But you
trees.
didn't. So that's how you measured ash dieback. For example, do you know the 2019 paper from Stocks
Yeah.
on ash dieback?
Yeah.
They use case control, right? Either
Yeah.
you have a ash dieback or you don't. In your
Yeah,
case,
in my case we used a gradual scale because we see it as a quantitative character. So we used a gradual scale, so in percentages instead of yes or no. Because to us the real life is not yes or no. It's a gradual, it's a continuum.
Okay.
So we assessed the crowns of the trees in the sites, and it was repeated measurements because we have many ramets per genotype. So we believe that our ash dieback measure should be a fairly good measure because we have this statistical, background.
No, that's good. I just was wondering whether you found similar SNPs than that Stock found, or
Actually not. I think it's very, very few of the SNPs that are overlapping with Stock's study. So that is also interesting. But then again, of course, I think the Stock's paper was basically on UK material, and we have like a continuum across Europe. So.
Okay. Yeah. Also, I don't know if you're here on the when I gave the first presentation, but there's someone in Bugs Labs that are actually looking at natural selection,
yes,
juveniles and adults. So
yeah,
it's probably something to keep an eye on. Cause
I think actually they already published it, right?
There's a preprint.
Yeah.
But
so
they've submitted, I think, Nature Ecology, but I think there's some overlap there, so it should be interesting to
definitely,
validate.
definitely.
Yeah. Okay.
So we have followed the work from Richard Box closely.
Yeah.
So,
okay. Cool. Thank you.
Hi, my question is for Ricardo. Ricardo, what kind of tree traits that are relevant for tree breeding do you believe we need to improve more? I mean, about growing, about defenses, roots, maybe. What should be the target of trees in order to improve our phenotyping capacity?
I'm sorry, but I was thinking another question. Can you repeat the first part of the?
What kind of tree traits that are relevant for tree breeding do you believe we need to improve more in terms of efficacy of?
Well, the question is that in some cases we're having a reduced number of traits in tree breeding, and it's challenging to include the new ones dealing with the adaptations. But the problem is to, as I mentioned, that traits related to adaptation are in some cases difficult to measure. We need a lot of genetic tests to have and then it's not easy to answer. They say, well, there are many different stressors, and my question is that in some cases we still lack some good traits to include in breeding targets. It's not a problem from the breeding perspective if we need to include this type of, for instance, in drought tolerance, we are using water use efficiency. Is because it's the best? No. It's because it's the only one that is easy to phenotype at a large extent, and they can provide some insight about the adaptation, but it's not the good one. Then this is, and concerning another point is that have been mentioned in the discussion that here we talk about a lot about major genes affecting some questions, but according to the traditional genetic theory, most of the traits are polygenic and according to infinitesimal model. Then if I repeat and GWAS in two populations, I expect that alleles are different because according to the model, there are many SNPs with small effects significant. Then for me it's not a problem. It's probably is related to the fact that this is a polygenic trait with many genes affecting. And if you change the environment, if you change the, and then this is also a lot it has a lot of implication in breeding. And then what? I have not answered, but because it's a really a very tricky question.
Good enough. Thank you so much.
Actually, I have a follow-up question about that, just very quickly, because it got me thinking. So I know that we hear a lot these days about how tree breeding programs require, and they require long-term investment. The tree life cycle is very long. So we're looking to new technologies for example, rapid phenotyping, or to be able to, get trees to produce seeds faster, or do things in the greenhouse, or different types of assays, or marker-assisted selection, things that can speed up the process. But in your opinion, do you think that we should still be paying attention to well-organized, efficient, comprehensive, traditional tree breeding programs and not lose sight in the value of those as we pursue these new fancy technologies? That's my question.
Well, for me the question is that, well, according to the condition is the breeding program, you need to adjust. Are you more interested in improving your phenotyping? For instance, in some cases in Southern Europe, we do not want to establish long-term crossing schemes for species that are really not so important. Then you can use genotyping technologies, genotyping tools to avoid this type of topics or for testing. Then there is a question that still that there is different papers concerning proof of concepts, how to use the information, but in my opinion the problem is that we need to test in forestries, and depending on the population that we are dealing with, or the stressor we are dealing with, which is the best approach. And I do not know which is, but the only question that we have a lot of advantages in comparison to previous studies that we are, now it's really easy to genotype in comparison to previous part, and the problem is that now it's difficult to phenotype because it's costly, but with the new approaches based on drones and so on, it's becoming really very, very efficient. And then the challenge is to incorporate this in breeding in breathing, in some cases, the movements are quite slow because we prefer to to base in traditional breathing breathing theory that incorporate very soon many advantages that we are not sure if they are going to work at the long term, at least in forestry.
Thank you to the speakers. And thanks, Jeff. Great. I think we need just maybe 10 minutes stop for having a coffee. Okay? And in the way, I'm going to explain you the plan for tomorrow, so you can be around and pay attention on your own. Okay? Just 10 minutes stop for having a coffee. Okay, I have to. Some 5 minutes for about the logistics for the trip for tomorrow. First, no, on Friday. Logistic stuff is if there is some people interested in coming back Porto because they are leaving from Porto, so. We will. To Vigo, we will organize the trip to Vigo to the airport, okay? Just please, the more operative stuff should pass over Maria and note your name if you need the trip to Vigo. Name and time, please. So for the field trip tomorrow, okay, this is the low pressure. You can see in this figure. It's the first low pressure in the season. Sorry, so, Rafa.
Okay, welcome to the last session. This is 4 talks about priming. It's a sort of a diverse set of tree species. We have oak, we have pine, and we have spruce. And first speaker is Christina from SLU, where you're a PhD student, right?
Yes. So, hello. I hope the coffee has kicked in, so you are awake again at the end of the day. So yes, I'm Christina Berger Neto, a PhD student from Sweden, and today I will tell you about traits underlying resistance in non-treated and Maya-treated somatic embryogenesis spruce. And in my, oh wait, I don't have the pointer. So the main idea in my PhD is to look at the plant's own defense trait and how we can use them to our favor within plant protection. And in the group I belong to, we mainly study conifers and one of their associated pests, the pine weevil. And the reason for that is because the pine weevil is one of the major regeneration pests in Europe. And well, yesterday we had 2 nice talks describing it from Raoul and Barley. So I'm not going to go into details, but to summarize, they like to come to the clear cuts and feed on the seedlings that we have planted there, the smorgasbord of seedlings. And they also like to girdle the plants, which cause a lot of mortality. So without proper seedling protection, they can cause a huge amount of damage and mortality. So they are a big problem. But well, there are many ways to protect the plants, but one way that has been increased in interest lately is the use of the plant hormone methyljasminate. It's a defense signaling hormone that you can spray onto the plants and prepare them for a coming attack. You prime them, so they are already yes. So they are already prepared when the attacking is coming, and this has shown that they have increased resistance against the pine weevil. And a few years ago, there was a study in our group that involved these Norway spruce seedlings that they are propagated through a technique called somatic embryogenesis, or SE. And this study kind of revealed a previously unknown plant protection advantage within these clones. And in this field study, it showed that they also had an increased resistance to the pine weevil, similar to the one from Maya, and they received about 30% less feeding damage. So, well, because of this, we thought it would be interesting to see what happens if we treat the SE plants with Maya. And well, what we wanted to know was if Maya would have an effect on the resistance on these SE seedlings, and we were hypothesizing that since the SE plants had already shown a high resistance, that maybe it was at its maximum, so perhaps it wouldn't be any higher resistance, or it could increase, but maybe not a lot, like a little, and we hoped to see some kind of increase. And then we used SE seedlings and non-SE, and the non-SE are just regular spruce seedlings, and we treated half of them with Maya about 2 weeks before planting them in the clear cut. And then we looked at attack rate, feeding damage, and mortality at the end of the season. Now I will only show you one of the results, because I have a bit short of time, but you are welcome to ask me if you want to know about the other results. And so this one is the total stem damage, and that means how much the pine weevils were eating on the whole stem or each plant. And then on the y-axis, you have the area debarked, and on the x-axis are the plant types. And here to the left are the ones not treated with MEIA, and to the right are the ones treated with MEIA. So if we first look at the no MEIA plants, you can see that the control had quite a lot of feeding damage. And here I also included the effect sizes, so you can see how much each treatment had compared to the untreated control. And as you can see, the non-treated SC1s had 32% less feeding damage than the control, which was similar to this previous experiment that I told you about. And if we then look at the MEIA-treated ones, the controlled spray with MEIA had even a little bit less, so 53%, while the most interesting one of them all were the SC plants treated with MEIA, which had as much as 86% less feeding damage. And this was quite a surprise to us, because even if we were thinking that maybe they would have some increase, we never expected it to be that much. So it's something really interesting going on there. And as you can see, it was also very little variation within those. So to conclude, both to the pine wheel. And so because of this, I mean, both because of this and because of the reason that the SC plants have been showing increased resistance also by themselves. The question is, of course, why are they doing that? And to shed some light on that, we are trying to do some further experiments, and some of them I will show you now. I don't have finished results from them yet, but I will show you a little bit so how we did it and some hints of the results that we have. And what we decided to look at first was the stone cell abundance, the chemical defense, so both the terpenes and the total phenolics, and then also the resin ducts. And that was the size, the number, and the density. And so first, the stone cells. So what we wanted to know was if the abundance of stone cells differ in SC seedlings compared to the non-SC ones. And stone cells are physical structures. In this case, they would be in the bark then, and they would make the bark more difficult to chew on. So because the SC plants had less feeding damage, we were thinking that they should have a higher abundance of these stone cells. And to show you how we did it, I borrowed this picture from Justin Whitehill. And we did similar to how they did. So we took 3 parts of the stem to cover the whole stem where we looked. And then we cut them in vertical slices and stained them, and then looked for these red structures. And this is what we saw. So the stone cell should appear in this. Well, I can show you. In these green parts, but as you can see, we found nothing. And I got a bit desperate, so I went out to the forest. I picked parts from wild spruce just to see, but nothing. So I conclude that Norway spruce don't have any stone cells. At least I haven't found them. But yeah, so I mean, of course, it's very interesting in itself. Not really what we were looking for, but yeah, so we kind of moved on. Then we wanted to look at the chemical defense, if it differed in the SC seedlings. And we expected SC seedlings to have a higher concentration or a different composition of these secondary compounds that make up the chemical defense. Yes, and this is how we did it. So because of these previous results, we chose to include MEIA also. From the beginning, we were only thinking of looking at SC. So half of them we treated with MEIA, and we did it twice. And then we waited several weeks for it to have an effect. But then to make sure that the plants were really properly induced, we also did some mechanical damage with a type of needle tool that we kind of pierced around the stem, and we also did that twice. And then we sampled both bark and needles, and the bark we took from this lower part where the pine will normally feed. And then finally we extracted these compounds in hexane and methanol. So the terpenes are extracted in hexane and the phenolics in methanol. But something we saw when we had extracted was that in these extracted liquids there were a lot of differences in color, especially in the methanol samples. And I mean, I'm not a chemist, so maybe this is normal, but I think it kind of indicates that there is a difference in extracted compounds. Exactly what, I don't know, but the samples are right now being analyzed in GCMS by our collaborators at the Mid-Sweden University. And, well, what we see very preliminary is that there are differences between the treatments, but we don't know yet what kind of differences there are, so that we still have to wait for a little bit longer. But I also hope later that I can see if there is any connection between the colors and the results from the analysis. And then for the last thing, we wanted to see if the size and the abundance of resin ducts differed in the SC seedlings compared to the non-SC. And, well, in the field, in this first experiment, we had seen that both the SC seedlings and especially the SC seedlings that were treated with MEA, they had a lot of resin. It's like covered with resin on the stems after the pine we had been eating. So we were thinking that there must be something causing this. So we were thinking that these SC seedlings, either they should have more or larger or a higher density of resin ducts compared to the non-SC ones. And this is how we did that. So again, we treated twice with MEA because we again wanted to include the MEA effect. Then we cut the stems and fixed it in something called FAA, and we cut this bottom part again where the pine will normally feed. And then we sectioned them at the same, well, about this area where the arrow is. And then they are stained with safranine and Astra Blue, a mix. And this is being done right now, but we have some preliminary, well, we have some slices of each treatment done, and they look something like this. So first we look at the constitutive resin ducts, and in the bark they are here, quite big, and there and there. So this is a non-SC, and there's an SC to the right. Sorry. And then we also look at the constitutive ones in the xylem, in the bark, no, the wood. And they are a bit smaller, as you can see. And then we come to the interesting part again, because when you treat them with MEA, you get something called traumatic resin ducts, and they appear here. As you can see here is a nice color of part. And this is the non-SC one with MEA, and this is the SC one with MEA. And like I said before, we were expecting the SC ones with MEA to have more or bigger resin ducts, but this one has, the non-SC one has quite big and many, while the SC one has small and quite few. Even some of them look more like this, so I can see 1, 2, 3, so there's very few. And yeah, this is kind of the exact opposite of what we expected. So I think this just shows how complex these plants are, because they always keep surprising us, which is of course fun, but confusing sometimes. But like I said, this is only preliminary, so we have to look at all the parameters and in all the samples before I can give the true result. But yeah, so this is how it looks like right now. And that's all I wanted to say, so thank you for listening, and thank you to my supervisors and my assistants and the people financing this. So, thank you.
Thank you. That was perfect timing. And next speaker is myself. So somebody can keep track of the time. Okay, I'm Paul Krokne, still. I'm from the Norwegian Institute of Bioeconomy Research, and I will talk about defense priming in Norway spruce, and also about inducible defenses in Norway spruce. It will be a sort of a summary of 20 years of working on inducible defenses in Norway spruce. The background photo is sort of a fun photo, because it was Melissa Magerø and I, we were writing a paper for 10-year-olds about priming of spruce trees. So if you can't follow my quite simple talk, then I refer you to this paper. It's written for 10-year-olds, and I think it actually will work really well to explain, for instance, bosses, administrators, what I'm working on. Yeah, and well, just a little Norway commercial. I'm the only one here. It's a pretty spectacular country, northern Norway, the mountains, but where I am, it's more like the dullest part of Norway. And that's where the institute is, in the southeast. It's a pretty big institute. We are 700 people, and we are actually all over the country, which is interesting. The one in the north, it's literally 200 meters from the Russian border. Okay, so I will very, very quickly say something about plant defenses, and then I will come with some examples of normal inducible defenses, and then priming of defenses. And all my examples will be from Norway spruce, and despite the title, I will just have a little bit of the underlying mechanisms. We're not really very. Why I'm studying inducible defenses in spruce is the Norway spruce bark beetle, the spruce bark beetle of Eurasia, Ips typographus. It's a real tree killer, and it's shown what it's capable of the last 5 years. It's quite amazing. It's almost like having a mountain pine beetle in Europe, probably maybe 300 million cubic meters killed. And as you probably know, it seems to get a lot of help from some associated fungi that will increase the virulence of each beetle attack. Yeah, and a little intro to plant defenses. This figure is in an upcoming book chapter with Justin Whitehill and Jörg Bollmann and myself. It's just a classification scheme for plant defenses. I sort of grew up reading about constitutive, inducible, chemical, physical, etc. I guess the idea here is that you can sort of put anything in the middle here, and then it can be any defense can have a direct effect, and it can be chemical. So I thought that was quite neat. And spruce defenses or conifer defenses, of course. There's been a lot of work on both constitutive and inducible defenses here, showing constitutive resin ducts in the sapwood, not really that many. And here is a ring of traumatic resin ducts. So it's really, as Christina showed, it can be pretty massive. And also there's been a lot of studies, of course, on particularly terpenoids and phenolics in Norway spruce. So the first example is a nice study where we could show quite clearly that that inducible defenses seems to have more sort of explanatory power when it comes to whether a tree will be resistant or not, than compared to constitutive defenses. So if you want to sort of phenotype a tree, here with experimental design was, this was not with beetles, but with the fungi that the beetles carry. So we inoculated trees, sampled them 2 and 5 weeks later, and then we induced beetle attacks. And then we saw what happened. And then the sampling was, what we did was quantifying terpenes. So we see here that the susceptible trees, they have a nice, I mean, maybe like a 70-fold increase in terpene levels. It looks quite good, but everything is relative, I guess. So if you compare with the resistant trees, the ones where the beetles couldn't really conquer the tree, you see they're more like close to a 300-fold increase. So the more inducible the trees are, the less suitable they were for the spruce bark beetle. And another really nice study by a Swedish PhD student, Christian Schiebe, was he actually went out in a Swedish forest during a bark beetle outbreak, and he sampled lots of trees, Norway spruce trees, before the beetles had been flying. He tried to think like a bark beetle, like an aggressive bark beetle, and it's really turned out to be really, really hard. Maybe 200. And then the stupid beetles, they only attacked something like 17 of them. So it's really hard. Even if you go along forest edges, and it doesn't. But he got exactly the sample size needed to get significant results. And so what he did was he sampled the trees, and then some were attacked by beetles and survived. Natural attacks, no pheromones, and some were attacked and killed. And if you compare those two, and these are just mono, sesqui, and diterpenes, so they're all more or less telling the same story. There's a bit of a difference, but it's not significant. But when you compare, this was the constitutive terpenoids, and if you look at the induced terpenoids, there was a much larger difference, and again, it was significantly higher in the resistant trees or the surviving trees. So somehow the beetles can immediately land and sense something about inducibility that we have to wait 5 weeks and do a lot of GCMS work to figure out. So it's we don't know anything about exactly what the beetles are doing there. But of course that's what they do for a living. I mean, if you pick the wrong host, you might die. So it's a very strong selection for making a good choice. And then over to methyldiazepinate. This has been actually, I guess this is one of the first papers on methyldiazepinate in conifers, done by colleagues at NIBIO and elsewhere, and just showing that if you treat a tree, we spray it with water, nothing happens, spray it with methyldiazepinate, and then we inoculate it a few weeks later, you have this massive ring of traumatic resin ducts. And a lot of resin coming out of the wounds, whereas here there's also wounds but no resin. And the first years, we didn't call this priming then, but and we used the fungal inoculation instead of bark beetle attacks because it's easier to deal with the fungus. And here, 5 clones of Norway spruce. If they're sort of naive trees and you mass inoculate them, they all die. But if you spray them with methyldiazepinate on the outer bark some weeks earlier, I guess 4 or 5 weeks, they all do very well. So they have it in them, they just need a little bit of early warning. And this effect, this is another fungal inoculation study, it just shows that we treated them in 2008, and even 3 years later, there's still a significant higher resistance in the methyldiazepinate treated trees. But it seems to be sort of smaller and smaller effect over the years. And just a fun thing to relate this to bark beetle behavior. It's when we let the beetles colonize stem sections that are, and then we quantify the main pheromone component. You see over 6 days in the lab, you see the beetles emit a lot of pheromones in the control, and hardly anything in the methylgesminate, sort of the primed or vaccinated trees. So that was a pretty cool thing. And also in a study with Nadir here, we saw that beetles colonizing methylgesminate treated bark, they were less attractive. So, I mean, we did this first, and then we went on to do this. So just showing that it has maybe some sort of unexpected effects on the beetles. And well, for many, many years, I thought that methylgesminate was an inducer of tree defenses. Well, I also showed you with a traumatic resonator that it is. But the confusing thing is that it's both an inducer and a primer of tree defenses. So when you look at the monoterpene levels, if you spray a tree, wait 4 weeks, take a bark sample, and do the GCMS, this is what it looks like. So we thought, well, 3-fold higher monoterpenes, that's nice, it's significant, but I mean statistically significant. When you look at the individuals, there were some trees that didn't really behave, and if you look over here, there's hardly anything going on in some of them, and some react very quite a lot. But still, it's only a few-fold increase, so it's not really very impressive, and maybe something should have told me that this is not enough to explain the really good vaccination effect that we see. But it took some years, and then we did things like that you spray the trees with methylgesminate, and then before you sample them, and then you sample them, but also you challenge them or you wound them just before sampling. And then there's a completely different picture, because this is what happens with total monoterpenes. Okay, so control and methylgesminate, in this case, I don't remember exactly, that this was probably just a single clone. And you see there's no induction by methylgesminate. So it's not really anything is happening. But then if you wound it and wait 24 hours, you have this 60-fold increase. So this is what the beetles are facing. So this is why it's so effective. So it actually, well, it primes defenses. So the general model is you do something, you treat them with methylgesminate, maybe you have a little transient increase in maybe terpene levels, but then it goes back to normal. And then you have a triggering stress, a bark beetle attack, and then you have this massive upregulation of inducible defenses compared to a naive plant that hasn't been having this warning signal with methylgesminate previously. So priming is sort of a long-term, low-cost version of inducible defenses. And there's some papers sort of telling you, there's some confusion around primings, and there's a few papers sort of telling it's like how to study priming. And you need to show memory effect, of course, low cost, stronger defenses, and better performance. And it's not really, okay, well, we'll see. So of course, there's some time between priming and triggering, and so that's the memory, and then there's a low cost, as I've shown you, and then it's a stronger defense, and then in the end you hope to see a better effect of the beetles or the fungus. So we did this in a field study in Sweden, and this was, I think a talk yesterday showed that if you treat with methylgesminate, you actually attract the insects. We also did that in Sweden. But the beetles land on these trees, but they hardly do anything. So it's a really good effect. And then we had a fungus inoculation treatment, and it also worked quite well on the beetles. Yeah, so low cost. I don't have to go through this again, I think. And then again, long cost and the stronger defenses, as I just showed you, just at both at the metabolite level and the gene expression level. And better performance. Well, we had in all our 20 trees in this study, there was not a single beetle egg in any of the 20 trees, whereas the other trees had lots of egg laying and larvae and pupae. Yeah, so better performance again, as I just showed. So inducible defenses can be prime. Could be a combination. Paper to be part of. I learned a lot from writing this together with these are hardcore Arabidopsis molecular plant pathologists, and this is Melissa Marre, my colleague. So it's like a nice overview of plant defense strategies. And well, this is my little sort of flow chart of defenses, and I think I'm out of time. The underlying mechanisms, I won't say much about them. It's just we've done some RNA-seq, and it's published in. We have actually now a series of 3 papers sort of going deeper and deeper into the underlying mechanisms in plant cell and environment. Finally we found a nice journal that wanted to publish spruce work, so we just keep on sending it, and well, there's some nice editors there. Yeah. And maybe I'll skip this complicated one, and then I thank you for your attention. Next speaker is Rosa. Does it work? Yep, you're a postdoc.
Quite short. So, good afternoon, everyone. Now, this afternoon, I will explain the second part of our project about chemical-induced resistance against powdery mildew using oak seedlings. So, as Paul just explained, the priming of the defense. This means that after an stimulus that can be different pathogens, root microbes, volatiles, or chemicals, the plants can respond faster and stronger to the subsequent attack. Our research is focused on 3 compounds, chemical compounds, 2 plant hormones, salicylic acid and osmonic acid. The first one related to a biotroph defense response, and the second one to necrotrophic pathogens. And the last one, BABA, beta-aminobutyric acid, is one of the non-protic amino acids that in different species can cause resistance. So first, as oak is also a plant, and these 3 compounds can create this induced resistance to different pathogens. First, we treat these oak seedlings with the 3 different chemical compounds, and then after 1 week infect with powdery mildew. To do the scoring of the oak seedlings, we use this scale between the healthy leaves and the tissue collapse, depending on the percentage of leaf covered by the powdery mildew, the white mycelium. In the first graph, we have comparing the control treatment. That means that we treat with water, salicylic acid, osmonic acid, and BABA. First, the early response at 7 days post-inoculation with the number of colonies per leaf, And the second one, the disease ratio, is with the disease severity ratio, translating this categorical scale to a numeric one at a 30 days post-infection, 1 month after the infection. So in the first case, we can see that initially both salicylic acid and jasmonic acid cause a little bit of susceptibility, especially when we compare with the water without any chemicals. However, when we let the disease progress, we obtain really resistant phenotypes with the beta-aminobutyric acid, then the salicylic acid, and a great susceptibility with jasmonic acid, less than the control. To see if this jasmonic, I said this before, just to see if this chemical treated in this resistance have a cost on the plant growth on feed. We recorded at the time the growth of the stem, the number of the leaves, diameters. In these cases, we focus this part on the baba and salicylic acid. There are the chemical treatments that cause their resistance to powdery mildew. You can see that growth lost. In the case of the salicylic acid, we need to say that also we can see this spot that appears like the programmed cell death. That can be a direct response, not the priming. So in this case, just to see if it's priming of defense of direct response, first we have the resistant phenotype in terms of the direct resistance with the reduction of the growth that we can see with baba, the cell death, the hypersensitive response that we can see with salicylic acid, and then to test the priming response, we made both callus deposition and the expression gene of the pathogen-resistant one gene. In these cases, the callus deposition with the treatment of the baba start to appear at the 2 days post-infection. We have here the control with water treatment and the great callus deposition that they then appear with the salicylic acid. However, with the salicylic acid, we got this priming profile of the PR1. When we compare the effect, the resistant phenotypes treated by salicylic acid and baba, we can see that the mechanisms are totally different and really specific to each elicitor. Just to disentangle these different mechanisms, we performed a metabolomic analysis. For this, we treat 3-month-old seedlings with baba by soil drains and the control with water, the salicylic acid and jasmonic acid by spraying. This treatment were 1 week before the inoculation with powdery mildew spores. And we collected 3 times point at 0, 1, and 2 days post-infection. These times were selected based on the callus deposition and PR1 gene profile. The first result of the metabolomic analysis were that a total of 3,387 putative masses present significant differences. At 0 days post-infection, 1 day post-infection, and 2 days post-infection, combining the 3 times point, we got more or less a separation between the treatments and also the healthy and the infected ones, with a really, really low percentage of the variation explained by the components. Due to this high number of differential masses, we decided to reduce the number, first using the water control to do the fold change and keeping the threshold higher than 2 and less than -2. we did this Venn diagram. In this case, is the most remarkable effect with baba, that also without any infection, because the mock plants were without infection, caused like a huge increase of the number of the masses that are different. For the powdery mildew ones, we made first with baba, this the same threshold with the fold change, and then we compare the mock of each elicitor with the powdery mildew of each elicitor. So this number represents the specific masses that are different when we compare with their respective mocks plant. Okay. When we saw the time course of this resistant response, we could see that is also at time-specific ones. The baba, since the beginning, is causing a huge number of changes in the metabolome. With the salicylic acid, we start later, at 2 days post-infection, respect just 20 that are different. Also, it's interesting the number that are sharing between 1 day post-infection and 2 days post-infection, like 100 times more with the baba than with the salicylic acid. Maybe it's also due to the low number of the masses that are changing at 1 day post-infection on salicylic acid treated plants. So with this, we selected, after this Venn diagram, continue with the analysis with these ones, the specific due to after the infection, related with the putative prime response. Using the Marvis tool, we proceeded with the pathway analysis, separating the baba prime masses and the salicylic prime masses, with both most of the electrospray, negative and positive. In this case, we got like the different pathways altered by the baba-treated plants, mainly alkaloids and flavonoids with the negative mode, lignin and aromatic compounds with the positive one. With the salicylic acid, we got a really, really low number of identified metabolites, so all the analysis pathways were not conclusive, with just 2 identification points. With the baba-specific ones, we selected a total of 17 tentative identified compounds at MS1 level to continue with the analysis at MS2 levels that were related with the resistance in other plants. Here is like these 17 compounds, majorly the alkaloids and flavonoids, with also the nicotinic acid. Our next step with these compounds is just to treat specifically the plants with each individual one and see if we got also the resistant phenotype. Especially because when we treat with baba, we got like more than 500 masses that are changing without the infection, so just to try to reduce the cost for the plant. Our future perspective is do also the transcriptomic analysis using exactly the same samples that we analyzed by the metabolomic. The second one is testing these same elicitors under field conditions using the biphorphase. The third one is try with other biotic stress, like the acute of decline, infecting the plants with the 3 genus of bacteria involved in this disease. Here I include an image of our first trial, that we can see the non-infected plants versus the bacteria infected ones. We got also more or less with the baba treatment a slightly resistant without this huge damage, and again with the jasmonic acid, the huge susceptibility. So the take-home messages are trees can be primed successfully by chemicals, different defenses and responses with salicylic acid, jasmonic acid, and BABA treatments are involved. BABA produces greater and earlier metabolome changes than salicylic acid. Salicylic acid and BABA have different mechanism responses. Preliminary pathways enrichment analysis were weak due to the low numbers of identified compounds. This putative identification of some compounds are associated with the BABA response, some of them with known defense activity. Finally, thanks to my research group with my supervisor, Estrella Luna Diaz, the different students that were involved in these different trials, and Victoria Pastor that helped us with all the metabolomic analysis. And of course, thanks for your attention.
Thank you. That was exactly 13 minutes. Leda.
Something.
Hello. So hello, I'll try to be short. In conifers, and I guess it's not the result, but it should have more or less a question mark here. So basically, where we are trying to look in this study is if actually this effect exists. And this was something that we started many years ago. Actually, to be true, Rafa and Luis started, and I jumped in the middle to analyze some of their material. And so basically, the concept behind that is that we're working with trees, and those are stationary and long-lived, and because of that, they are sort of endemically exposed to some herbivores and pathogens, and they also some of them have a sort of relatively limited seed dispersal. So quite a sort of attractive idea is that it would be very nice if the sort of, because this, their offspring is going to grow at the same conditions as these mother trees, it would be sort of very nice if the trees could sort of pre-adapt their offspring to this environment where they are growing. This is a very attractive idea, and of course one has to think in whether that is theoretically possible, what could, or at least how trees could do that. And there are several ways that in principle it could be possible, and some of them would be like, for example, by packing the seeds differently, so changing the composition of the seeds. The other way would be perhaps with some epigenetic signals that would be transferred to the next generation. And also, we can really think in many other ways. You can also help your offspring with your microbiome, and also directly or also indirectly. You could even, thinking as a pathologist, you could be spilling pathogens to competitors, so your offspring could be better. So there are many ways, and in this study, I guess what we wanted to focus was on the first two mechanisms. But I'm just saying that there are many ways that this could happen. So what Luis and Rafa did was to prepare a very nice material to test this hypothesis, I must say, and this was done by preparing a full SIP experiment. Why is full SIP? Because it was, I controlled both the maternal procedents and the paternal procedents to create some offspring, and this was done by doing 3 controlled crossings, with the only difference is that these 3 controlled crossings were repeated 3 times. So the mothers were stressed in different ways. So we would have some mothers that were not stressed, and we would have some that would be subjected to mechanical wounding, and some that would be subjected to methyl jasmonate, right? And that cross with the same parent. So basically just to, I know it's almost 5 now, but just to repeat the experiment. So we had like crossings, and again, it was the same genetic material. The only thing that was different was the mothers were stressed in different ways, and of course there were some that were not stressed. Okay, so this is how the setup looked like for the trees. I wasn't there, so this is a picture that they gave me. And also in this experiment there were other treatments included. So, but yeah, we'll mainly focus on these two treatments, on the methyl jasmonate and mechanical wounding. So we were not interested in what happened to these trees, but what happened to the offspring. So cones were produced and seeds were collected, and then we looked at what happened to them. So already from the beginning, to me it was quite clear when I got the data that the things were happening already at seed level, sort of. So we look at the, there were differences between genotypes in how many seeds were produced, but already some sort of things were happening in the maternal environment. So there was also changes already in the, for example, in the seed number, and this was not significant, but in other sort of traits related with the seeds, we could start seeing some significant effects. For example, there was here again with the amount of them that would germinate, so we could see that there was a higher germination, not being stressed. Slightly different, sort of an effect in how these seedlings look like after this, they were germinating. So we had some, if you look at the phrase weight, there was again an effect of the genotype. Would produce small others. So this was saying how the seedlings would look like, and we wanted to go a bit deeper and see whether they would be sort of more or less prepared to cope with this stress that the mothers have suffered. So we designed a very simple sort of herbivory infection experiment, and as I mentioned, we had this sort of source of material, and then we subjected some of the seedlings. We had sort of a time series of methyl jasmonate, so we will have them growing without nothing, and then sample at 3 hours and at 6 hours after applying methyl jasmonate to the seedlings. Again, here there was a sort of, we have to discuss what to do, but basically most of the experiments we focus on this genotype that had sort of produced more seeds, and we felt that maybe there was not so much bias on the seedlings. So we did lots of experiments, and sort of starting with the RNA-seq experiment, then we took also this candidate gene approach, which we did with qPCR. We also tried some gene body methylation analysis, and finally we did the bioassays. And now looking backwards, I think we should have done like Christina and start with the bioassays, but anyway. So we started with that, and this is how the plants look like. So you can already see there was some variation in how they looked like. So when looking at the RNA-seq data, a bit in connection with Paul explained, of course, when you subject the plants to methyl jasmonate, they immediately react, and you can see here, this is so how they are, and then after 3 hours and 6 hours, you can see like lots of changes in regulation. But we were not so much interested in how these plants were changing from here to here, but whether this change was different depending on those plants had originated from different maternal environments. So that was the sort of our goal. And so we started looking at how they were expressing genes without any methyl jasmonate. So basically we compare methyl jasmonate treatment in the mothers, mechanical woundings in the mothers with no stress in the mother. So how do they were at constitutive level, the seedlings? And we could see lots of genes that were differently regulated, mainly of them with involved in this resin synthesis and PR proteins, also phenylpropanoids. Many of them were sort of located at least in the membrane. And what was, I think, interesting was that seedlings coming from mother stress with methyl jasmonate or mechanical wounding, they shared many of those differentially expressed genes with the control. And for example, if you look at this, the gene that has the higher expression, in both treatments it was this longifolin synthase, which is related to some resin synthesis. So it looked like there was some common, at the end of the day, I'm sure the mechanical wounder was eliciting some similar response. And then when we looked at later, so we compared like, okay, so how the constitutive versus after methyl jasmonate in these 3 ones, and then we look at these genes versus these genes, sort of, okay, I think you understand what I mean. We again saw that many of so there were some differentially expressed genes, and methyl jasmonate treatment in the mothers and mechanical wounding in the mothers, and there was also many of them were shared, so it looks like there was almost the same stress in a way. We also took like a candidate gene approach, and we tested many genes in qPCR. This was done at the time that you needed sort of to validate your findings with RNA-seq. I'm not so sure you have to do this now, but anyway, we know we had some good primers and some genes that were well annotated, and I'm just going to focus on some set of genes in the phenylpropanoid. Also because I'm more interested in those perhaps, but we had like a set of genes that we know that are involved, for example, in this necrotrophic interaction, and this is Norway spruce, and you know, these trees, they sort of compartmentalize the pathogen with these reaction zones. We know that these reaction zone formation is activating many genes, and so we tested those in these seedlings that were coming from these different maternal environments. And just to remind you the time series, so we had like at this constitutive level, and then 3 hours, then 6 hours after applying methyl jasmonate. And you can look here, the gray and the black, these are the different maternal environments, and you can see that, for example, Paul or Chalconti, Sumerase, or many of those genes, they show a really higher expression than the control maternal environment after 3 and 6 hours. So there was some sort of like a transgenerational priming to this application. So we were quite happy, and perhaps we should have stopped there, but then we did the bioassays. And that was a mistake. But of course we are interested in the truth, but this was what happened. So we did, actually, this was a lot of work. We did a big bioassay. Well, I shouldn't say we, it was Rafa and Luis who did it, and there were no differences for how the seedlings would react to Hylobius, so it was exactly the same area that this guy would eat. And also we did some inoculation experiment with inside to do a qPCR. There were also difference. As conclusions of this work, so I would say that the value is that I think we can be sure that the experiment was well done. So it was really done with some cool material, fully replicated, and different genotypes. And we could see that there was an effect with the maternal environment, and possibly seed provisioning, and already in this response to methyl jasmonate. This was not an effect that was common across all the genotypes, so we could see variation already from the start. We could see, for example, an interesting result in priming in genes related to the defense to heterobasidion, but unfortunately not so much effect in the bioassays. Anyway, thank you so much, and if you have questions.
Thank you. That was your time. I'm not sure if I'm sort of organizing the discussion. Yeah. So I step down.
Please, the speakers. You can go.
Hey, okay. Priming is, okay, I'm Blessing from the University of Göttingen. Priming is pretty new to me, so it might be a very easy question, so any of you can answer. If you prime, it means that it induces defenses sort of early. So let's assume that you prime seeds or these seedlings. I saw from results that after a few days, then it makes sense, but does it wear off later? So if you sort of prime the seeds or the seedlings and you use them for, you establish them on large areas, does it still do the work after a long period of time? Or do they wear off at some point?
Well, what we've done in Norway spruce, it seems to, it wears off, but and then what's long term? In Arabidopsis it's like 11 days, and it's spruce it's maybe 5 years or 3 years. So it depends a lot on the plant. But I showed some set of results showing 3 years. It's still something going on, but if that's the primed response or if it's the continuous upregulation, we don't know. So I don't know if anyone else.
We have some studies with tomato, for example, with Stuel and Katie, and they can see some effect also between generations. You treat the mother, and for example, the first progeny is also resistant to the pathogen. With oak, the maximum that we treat is like left 1 year, and at the second year, the only survival under field condition with the powdery mildew, there are the oaks treated with baba. So at least with oak, we can see at least 1 year of effect.
Okay. I wanted to ask a question to Jonas. Very interesting presentation. You've done a lot of work. I don't know if you have any potential explanations for your results, but once someone said to me, RNA panics, but proteins don't. Do you think that might be a helpful avenue for interpreting your results?
Yes, well, perhaps I'm not too sure we want to do much more experiment with that, but certainly we were expecting some sort of phenotype there. I don't know exactly why we didn't. Perhaps we have thought that maybe the trees, because this first experiment we did with methyl jasmonate, there were seedlings, they were quite small, but in order to do the inoculations that we grew a bit more the seedlings. So maybe part of this sort of small priming was sort of erased by some other, like, I don't know, environmental factors, right? So that could be one of the reasons. But yes, absolutely, maybe we should have done some metabolome, but yes, at the end of the day, what I mean is that we can find some results with even with metabolites, but if at the end we don't get any results with the bioassays, then it sort of lose a bit the power of these results.
I just have a comment on using Hylobius. I guess it's like a bulldozer, and it's not really caring so much about. So maybe it's not. Maybe it's a difficult bioassay species to use because it is this bulldozer, and it seems to, not be very sort of discerning between stuff. So I don't know. But of course, you also did inoculation, and it didn't work out either.
Oh yeah, thank you. This is the question for experiment with Baba. That's a very significant result. I can see the induction. I wonder, have you measured the metabolic cost of such a high induction within a day of spraying, plants with Baba? Just identifying, within 2 days, what several hundred chemicals that must have some metabolic cost. I don't know what it means, but I'm not sure. Are you investigating that aspect?
Well, we treat with the lister like 1 week before, just 1 week before the infection. Yes, we thought that we need like the first curve, and then that all the metabolome reaction just due to the chemical stop and then go to the baseline, for example. That should be the real priming. The cost for the growth, we started to measure since the germination, because all these oak seedlings were germinated under control conditions, more or less. So we started with the same provenance, just to reduce the variability, just because the oak is really variable between them. We use 10 replicates per each treatment. Well, multiply by all the mocha plants, the infected ones, and keep with the chemical ones after 1 month, more or less. The really long experiment was 6 months after the growth, and at 6 months you can see the real damage with the growth. At least more or less between 5 or 6 notes of difference between the BABA treated ones and the water one. It's true that, for example, with the inoculated ones, the problem that we got was that the control and salicylic acid continue producing leaves. The problem with these leaves is that they are too young, they are too susceptible to powdery mildew, so finally they fall again. And we need to wait this year just to see that the BABA start with lower height initially, but bigger diameters, and also produce the second growth, the corresponding to the new year. That with the control with the water or the jasmonic acid is impossible because they dead. They can't survive to the second year after the powdery mildew infection.
I just want to share my observations, some of your data. I'm sure you noticed that too. Day 1 and day 2, your BABA, there is some overlap of some chemical produced by you, but your assay application, there's only one overlap from day 1 and day 2, suggesting that plants really start picking up more and more chemicals that start producing metabolites. I found this very interesting.
Thank you.
Hello. So my question is for the general, to all speakers. It's a general question regarding the session. So we did see Paul's work involving priming that took almost 2 decades worth of work, and then we do see Jonas. There was the problem with the bioassay where you could not validate what you found. So with this observation, my first question is, do you think it's just methyl jasmonate and jasmonic acid? Can you really draw the line and say, priming in trees is a jasmonic acid affair? And why not look at, I haven't come across any paper, but I'm curious if you know anything of why salicylic acid or its derivatives such as methyl salicylates, which has been used in some plant pathosystems for different priming responses or different defense responses. Has it been a failure in tree systems, or can it be used? And then, is priming a permanent effect, or can priming also be lost? So whoever wants to answer.
I think there was a lot of questions there, but well, I think methyl jasmonate is used because it worked, and there's not thousands of people working in conifer defense, so you just stick to something that works. And of course, methyl jasmonate is operating in spruce, so if you sample and you can detect, well, not methyl jasmonate, but this amino acid conjugate of jasmonic acid. So So it's definitely operating, and I think it's just used because it works, and it's non-destructive, and I think people have tried other stuff that maybe didn't work so well. And when we started out, we were working on 50-year-old spruce trees, and whatever you can get through the cork bark, you're just lucky that anything gets through. And we have no idea how much goes in there, but very little, I think. Yeah, at least that was a partial answer to one of the questions.
So regarding the priming question again, and this is for, I guess, everybody, but particularly for Paul, it seems to me, and I'm asking this because I don't know all the details, but it seems to me that you are presuming that there's nothing going on between the initial application and the effect. So, for a certain amount of time, like, you know, for example, you would treat with methyl jasmine, and then 4 weeks later, you would challenge, and you would see that in the absence of methyl jasmine, you have a little bit of an increase in terpenes in susceptible trees, but if they were treated with methyl jasmine, you get this enormous jump, right? So I'd like to know, have you really checked over time how the effect evolves in that interval of, say, 4 weeks? So, for example, do you check the response in the absence of a challenge? You check the response like at 24 hours, 36 hours, and so on, more and more dense.
Melissa, Sam Wilkinson, who was one of the lead author on this review paper, he did that on spruce more earlier after methyl jasmine treatment, a time series, and I think it's correct that it might be like in the model, but then of course it also depends on what you measure, right? Because we like to measure terpenoids, and but then of course the plant or a spruce tree that is primed or attacked, it will launch all sorts of different defense programs that run in parallel. So it is quite chaotic, and that was the slide that I skipped. It's trying to sort of illustrate some of all the things that goes on at the same time. So I realize that I've been thinking about that either they do this or they do that, but in fact they do all sorts of things at the same time. And different genotypes might do different things, but they arrive at the same goal, which is to be resistant. So I guess it's in reality it's probably quite variable. Yeah, but it's also because the fungus is still growing there, so it's like a continuous, it's an immediate and prolonged upregulation because the fight goes on. So that's I guess the beauty with methyl jasmonate is that you can do like a non-destructive, so at least you don't have to do any wounding. So so that's why it was interesting this field study in Sweden. We did both. We did fungus and methyl jasmonate, and so I think we could sort of point to both of those inducible defenses.
This is to Jonas. You showed this slide, but I didn't really catch everything you said on that slide when you discussed, you talked about defense against necrotrophs, and then also in relation to the application of methyl jasmonate, right? So, because we've done something similar to that where we kind of compare the methyl jasmonate produced, the gene encoding, the methylation of jasmonates. We kind of compared the expression of that gene with, to infection from Cornatia magnesia, which is a biotroph, and then how that is different to infection from a necrotroph, Rospana clavigerans, with also mountain pine beetle. And we saw that there is much higher expression for this gene against the necrotroph compared to the baotroph. So when you are priming with methyljasmonate, do you also consider the innate methyljasmonate produced within the plants?
I'm not sure I follow you because we didn't.
So you are priming, right? But there's also methyljasmonate produced within the plant, right? Just by infecting the plants with the necrotroph.
Yes, but okay, I see what you mean. So, no, but we didn't do how to say? We didn't do an inoculation with, and then we measured those genes. So I just showed like a selection of genes that we know they are in sort of involved in this interaction with Heterobasidium, but we didn't do qPCR following the inoculation with Heterobasidium.
Oh.
Sorry, I think it was my fault. I didn't sort of make it so clear there. It's just sort of the reason why we choose those genes, those candidate genes, because we knew that they would sort of they are important in the interaction with this pathogen.
Yeah, but is it something you guys might want to look at, like, because when you are dealing with necrotrophs, you most likely would expect methyljasmonate being produced in the plants, right? So
yes, of course, yes. So that's why we So in a way, when we wanted to see if those plants that had been primed or their mothers were stressed, how they would respond to a pathogen or pest, we simulated that sort of pest or pathogen attack by applying methyljasmonate to them. So you see what I mean? So that we would, at least for sure, we would get a response.
Yeah.
But I think, is this what you're saying, sort of? But we never did RNA-seq or qPCR following any inoculation of a pathogen.
Oh, okay, all right.
We just looked at methyljasmonate. We saw like some response. So maybe that would answer also the question before. So maybe we saw some transgenerational effect because we were doing this sort of huge hormonal sort of input into the plant, so that something happened. But maybe with the Heterobasidium it wasn't enough. You see what I mean? Or maybe with the Heliophis it wasn't enough, so to see that different.
Okay, thanks.
But thanks for the question.
Can you hear me? My question is from Christina. Very interesting presentation. I was wondering about somatic embryogenesis, because I know how difficult is conifers somatic embryogenesis, in general micropropagation. I was wondering how efficient was your system, basically the method of inoculation. And the reason is because 3-4 months ago I proposed same thing for spruce, for Sitka spruce in Canada. And we had some colleagues that they were against that proposal because they were discussing about efficiency of this in vitro method versus traditional method. So I had to defend it, and I tried as much as I could to defend the method. But then at the end, for now, they said, you do it with your budget. Basically, they didn't give me extra funding for that. But I was wondering, how efficient is your method in compared to traditional method of inoculation? And basically, did you establish your protocol yourself, or you basically used another author's protocol for micropropagation?
I mean, are you talking about the actual propagation of the acid trees?
Yeah, about the basically somatic embryogenesis, micropropagation of Norway spruce that you explained that.
No, because we don't do that ourselves.
Okay.
Yeah, we get them from, I call it a company, not a company.
So, so he's the...
Yeah. Oh, yeah.
Yeah. I'm Andreas from Skogforsk, and we are actually we produce the plants for your experiments, and that's why. But we have a small lab, but where we produce some experimental plants for SE plants, and it's really tough actually, because I know you ordered a certain quantity of plants, and I thought maybe you got 1/4 of it, so we had problems getting all the plants. Yeah, there was one study. They started with 1,300 SE lines, just putting embryos on plates. And then after, then they reduce it to like 700 cell lines coming, and then it's just going down.
So, well, Andreas and I made our PhDs together on somatic embryogenesis in spruce. So the initiation frequencies can be quite good in spruce if you are lucky and you catch the seed at the right time. Then you can reach probably something around, I would say, 60, 70, 80% of induction. And then, of course, you will have a drop-off when you look at proliferation and eventually then, as Andreas pointed out, germination. And the last experiment, which I'm kind of sneak peeking at while I'm talking here, we are having from the initiated proliferating cultures germination frequency between 30 and 70%, which I don't think is too bad.
But isn't it true that Swedish forestry is implementing SE plants, so you produce like 20 million or 100 million or something? And if that's true, then it can't be impossible.
It's not impossible, but I don't think that's going to happen either, because we have a cap on how much clonal material we're allowed to plant on land in general. But that's the general idea of that companies, I mean, Sweet Tree Technologies production pipeline is to produce clonal material from the very best lines and then sell them to forest owners that wants to plant them.
And also Finland, I think.
Finland have registered SC lines, which, I mean, material, which they sell.
Another species that we were working on somatic embryogenesis was eastern white pine and western white pine, and we had a colleague at University of Laval. She got like around 20 to 30% success with that one. So that's the reason we had lots of opposition against using new techniques, and everyone was pro-traditional, basically breeding. So that's I'm asking. We can continue our discussion later. Thank you.
I'm Jeff. I think you all know who I am by now. I have a question for Paul. I have a question about your study and slide. I have a question about a particular study and slide, but first I want to make sure that I understood it correctly because it was very interesting. So it seems that these Ips typographus beetles in Norway spruce, when they're out in the landscape, choosing or selecting the trees that they want to attack, they were able to differentiate between the ones that would have more greatly inducible defenses. But first I wanted to make sure I understood. So this greater level of induced defenses that was quantified in the trees that were basically remnants or survivors, this is interpreted as because they had higher levels, but could there have been a role of fungi that in the trees that were colonized by more beetles that led to observing like less? Maybe I'm misunderstanding the study design.
Yeah, I didn't explain it very well, maybe. But, well, Christian, he sampled, he took bark samples low, way down. I mean, the beetles, they don't attack below a certain height. So that, and then he did induced samples with methyldiazolate treatment, waited 4 or 5 weeks, and then he got the induced response. And then the survivors, they weren't really hammered by the beetles. They were not, well, maybe they were not the only survivors, but they were unattacked. He observed landing, so they were visited by the beetles, and there were no real attacks.
Okay, great. I just wanted to make sure I understood that first. So my question is, if there's variation, assuming it's genetic variation out on the landscape of trees, some of them have more greatly inducible defenses to bark beetles, and the bark beetles are able to choose, then when those trees that have less inducible defenses are removed from the landscape, just like in a thought experiment, and now you have this landscape of trees that is dominated by these trees with more greatly inducible defenses, the beetles are still, I mean, now they don't I mean, without the choice, they would be attacking these other trees and probably with sufficient populations still killing them, or, right?
So I actually doubt that the spruce bark beetle is a huge selection pressure in our forests. I mean, this is based on, I'm not an evolutionary biologist, but I mean, it's not very likely to be killed by the spruce bark beetle, I think. It's much more likely to be attacked by root rot. But so I'm not sure. And then there's this there's a random component to whether you die or not, because you might just live next to a weak tree, and it also depends on how you tolerate drought. So there's a whole lot of, I mean, usually the stress trees really stressed trees will be attacked by the beetles, and then others are just sucked along in the disaster. So I don't think we could expect that all the spruce trees should be then these very inducible ones, if that's was one assumption.
Sorry, I guess what I was getting at is like, is there a handle there for improved resistance? Or is this just something that beetles are making choices, and probably given the choice, this is fine?
At least I know that in Norway we breed for Norway spruce, and it's only about the productivity and wood quality. No resistance at all yet. And if they choose something, they will choose root rot and not the beetles.
Excuse me. I wanted to ask, what do we know of the underlying mechanism that mediates the priming response? Is it something like microRNAs? And would that knowledge be helpful in guiding future experiments? Thank you.
Yeah, we are trying right now, seeing with the tomato line, if this BABA treatment can cause some epigenetic marker, especially because we got this effect on the progeny, this really, really long-lasting effect with oak, because 1 year after the BABA treatment is a lot of time between them, and especially with the tomato, the only treatment that causes this long-lasting is if you treat really, really early the plant into a seedling stage. If you treat, for example, in flowering, you haven't any progeny resistance. So we are thinking that it's through epigenetics more than through RNA. Because, for example, in this case, we are seeing the metabolomic analysis with the line, gene, transcript, protein, then with all the post-transductional modification that we have like 3,000 masses that are different. Just treating with this, maybe it's due to the epigenetic marker.
Thanks.
Hi, I have a very fast question to Christina. So from the previous discussion about your materials, it seems like you are using for your SE experiments like random plants, right? These are not clonal that go through the SE and then you spray it and then you analyze them? Or are they all genetically identical?
You're talking about the non-SE ones now?
I'm sorry?
The non-SE ones.
Yeah, I'm talking about the plants that you then spray and then you analyze. I was wondering whether you actually account for your genetic variability to start with, or whether they are all identical, and then because then they differ.
Yeah, we have different families. In this experiment, it was 19 families with I don't know how many clones within those families, quite a lot. For the other experiments, we have 6 genotypes. So the problem is that the non-SE ones are not genetically matched. We try to get that, but it's not that easy. So, yeah, we have some kind.
So because the follow-up is, if that's the case, it indicates that there is like an overall one key to all of that, right? That improves your responses, and that there is like one underlying overall mechanisms that allows you to get more responsive plants, more resistant plants in the end.
Yeah, I mean, kind of what we have seen, at least in this first experiment with the 19 families, we looked a little bit of difference between the families. And there were maybe 3 that were differing a little bit, but most of them were reacting the same, so it seems to be quite similar among these families that actually, like I say, go through this process. So I don't know really what the selection is there, but they seem to behave quite similar.
I think someone was overlooked over here for 10 minutes. So I will... yeah, but I think it's...
You're not moderated.
Thank you. My question's for Christina. Your talk's amazing, and the results look incredible. I'm so excited to read the paper. But you talked about resin ducts, and it looks from this preliminary data like it's shocking that there aren't as many traumatic ones. So I've got like 2 follow-up questions. Did you look at resin flow or composition? Like, even though there were fewer ducts, did you see any differences in volume? Is the first one. And also, with the resin ducts, did you also challenge them, or did you just do methyl jasmonate? Because I know when you were looking at the chemicals, you had a needle challenge as well, but did you do challenge before looking at the resin ducts? Those are my 2 questions.
So for the challenge, we didn't do it for the resin ducts because we figured out it's not necessary, because it's like too little. Sorry? Yeah, I'm a little bit tired now, but we didn't do it because we didn't think it was necessary. Now I forgot why, but sorry. And then what was the first question? Oh yes. No, we haven't done that. That's actually something we're talking about. That if this is the final results, then it's kind of contradictory to how can I have more resin. So we were thinking maybe they produce resin like all the time, or I don't know. But that's one thought we had. So it would be definitely interesting to test. But, yeah.
Thank you. I have a question about all 4 of you. Under what biological conditions we can really see priming in nature? Okay, in my opinion, all the time we might see that priming. In my opinion, what the priming does, the plant have a different type of defenses. The first one is the immediate response, right? Happens within usually a couple of days or something like that you've seen in the field. There's a delayed response. I think that's why the priming does it. The tree or methyl jasmonate application helps the tree recognize the impact within a few days, and then plants are able to respond to it. I'm not sure you have any idea on that one. How do you incorporate your results, what's happening in nature? That's one. Second thing is, I think we have some good understanding of the pathways induced, biochemical pathways induced by methyl jasmonate or BABA or whatever hormone applications. Is it time to come to perhaps incorporating those things in plant genome, and then that could be next best thing after BT that incorporated in the aspen or poplar genome, that you get the giant trees? Is it possible? I'm just curious.
Will you answer about nature?
No, I think you can do it better. I don't know.
Yeah, well, priming in nature, I'm not sure. I mean, it's interesting and it's very potent, but whether it's important, we tried to do, we had failed bark beetle attacks in the Swedish experiment the year before. But then it wasn't a planned experiment, and it didn't really, we did some mistakes, so we could never really show it. But I think that failed bark beetle attacks, maybe early in the season or maybe the season before, could be, that's what we speculate, could prime trees, and that could be like one of several factors that contribute to outbreak collapse, but that's purely speculation.
Oh, I see impact of drought priming the defenses. Mild priming can have a positive impact on plant defenses in the summertime, early season, I've seen it, we noticed that induced defenses increase a lot after mild drought on the trees. Or some other pathogen impaction, mistletoe that we work with, they do prime. Defenses maybe host is, I will say, less suitable for the following attacks. Or it could be defoliators, I don't know. This is for bark beetles.
Yeah, and also we see if I walk into a forest and I try to prime a tree with metal jasmine, it will be very primable. I mean, even so, it's not like they're all primed already. So I don't think the whole priming thing is like an artifact, but it might not be super important. I don't know. Then I don't really understand why they have all these mechanisms, but because it's very clear.
Yeah, it's the last one for Enrico. Just a comment for that. This okay. That this a kind of thing. I think so. That the rising question is that this priming or sensitive to the attack, and this priming effect is the same in all the species, because we are looking at differences in priming within species, but we lack information about across species in conifers, in instance. And the second one is the somatic embryogenesis and epigenetic marks. What is the effect of the ontogeny of the plant, of the origin of the material, and what is the effect of the hormonal treatments during the production of the plant. So just ideas.
I just wanted to make an observation that I'm older than most of you guys here, and it's interesting to think back in the 90s when I was doing my first postdoc in Germany. People were talking about memory effects in trees, particularly Norway spruce actually, or Scots pine. They were also talking about delayed induced resistance. That particularly applied to insect damage. I don't know if you remember this, Paul, but it was the effect where feeding by insects this year would induce a stronger resistance to feeding the following year. In the case where I was working in Germany, there was this idea of a memory effect induced by air pollution on trees. So trees subjected to air pollution 1 year would be more resistant to other stuff the following year. So these concepts have been around for a long time, and it's just amazing that we're still trying to figure out how this happens. Just an observation.
Yeah, we have to finish. It's just.
There you go. How long have you been, and this is a question for all of you, and you might have said it, but it's 6 and my mind might be not too concentrated anymore. How long do you see the effects of prime? And so if you prime now, even with methyl jasmonate, how long after? It's 1 year, 2 years? Has anybody looked at how long this effect is maintained? Did they? I'm lost. I might be like, did they?
The answer was 3 years. But
okay, sorry. My brain was like probably very tired. It's 6:30. Come on.
Okay. Thank you. Thank you for this discussion. So it's time for the next activity. It's very close here. Just we need to walk like a little bit. The name is Maria? Okay, Maria already sent you an email. Please don't forget to put your name on the list if you want to attend the ride tomorrow. Just for ride, yeah.